the world who come over here to exchange experience and share ideas on the exciting field of life science research and their application. Today's webinar has received more than 450 registration requests with participants from various institutes in and around India, making this conference a truly an international webinar in spirit. Outstanding advances had been made in the past few decades in the life sciences and biotechnology, including genetic engineering, genomics, proteomics, and bioinformatics. Knowledge obtained in the field of life science and the techniques developed hold the potential for improving human health, welfare, economic development. Although biotechnology has created many opportunities and applications for public health, medicine, agriculture, and the food industry, this progress has important ethical, legal, and social implications. Research techniques and knowledge in the life sciences can be used for both legitimate and illegitimate purposes. Therefore, this raises the problem of how best to manage the risk associated with such research tech, uh, techniques and knowledge without hindering its beneficial application to public health and welfare. This webinar highlights the recent development in life sciences and its significance in science and society. This webinar aims at providing key inputs to the recent scientific contribution by different fields of life science research. In this virtual conference room, I recognize some familiar names of participants from government and private institutions and major scientific groups. We have all worked in this field of science, especially in life science, and we focus on the recognition and appreciation of life science as a major contributor in science and research community. French biologist Dr. Louis Pasteur said, if it is a terrifying thought that life is at the mercy of the multiplication of the minute bodies. Here he is implicating minute bodies as microorganisms. And he told that it is a consoling hope that science will not always remain powerless before such enemies. Now we all understand the importance of life science technology and innovation in our day-to-day lives and the way in which they are transforming the world. If this was not clear earlier, it is blindingly obvious now in the time of COVID-19 pandemic. Since technology and innovation are indispensable for our response to and recovery from the crisis, equally important is our collaboration in this great effort. Equally important are deployment of scientific invention, innovations, and research on a scale that will reach their target audience. I am sure we will also reaching out to many new trends and friends in life science. And this is not just another scientific talk, but the way to communicate, interact, and evolve and involve the young generation in modern approaches to the field of life science and research. I'm glad that we have got some prolific speakers for us to make this webinar an informative one. Ladies and gentlemen, I believe that by participating in this webinar, we are in the right place and right time. Together, let us accelerate the exchange of ideas and knowledge. I'm confident that you will be finding new ideas, fresh energy, and motivation towards this field. I wish you all a very successful webinar and brainstorming session ahead. Thank you, and I once again welcome you all on behalf of the Department of Zoology, the Kamdev Autonomous Region. Now,
এটা ছাড়া। Welcome. Registration from art କରିଦେବି ଏବେ ହବ ଚେଞ୍ଜ କରିଦେବି ମଡାରେଟ ପତ୍ର ବାନ୍ଧି ଶୁଭୁଛି ନା ଦେଖିଲ ମୋ କଥା I'm very sorry there was a disturbance in net connection. Sorry, gentlemen. Uh, ଶୁଭୁନି? So we have received a large number of registration from participants around the world. We are proud to have you all here at this important virtual meet on a global platform. Participants around the world will discuss today on this vibrant and intense, and they will exchange in views, knowledge, and experience. On this occasion, we are honored to have Dr. Sujit Pujari from USA, Dr. Uh, Sujit Bhutia, Dr. Ishwar Bhaitharu, and Dr. Sanjay Kumar Sahu from USA. We will have Santosh Panda from NIT Rourkela, from Washington University, and Sujit Kumar Bhutia from Department of Life Science, NIT Rourkela. And in this occasion, friends, this webinar is taking place at a time when a slew of historic reforms in world are laying the foundation for greater formalization and digitalization, creating the right institutional milieu for various organizations to collaborate together where the whole world is facing a situation like COVID-19 pandemic. Many issues which have had tempered mankind in the past are fast fading away, providing a much larger canvas for the instrumentality due to rise in the field of life science research. Widely called as biology, life sciences include all areas of science that might include the scientific study of plants, human beings, animals, and also their vital processes. Life is all around us, which all includes tiny germs crawling over land, water, and life science analyzes the origin, progression, expansion of life in all forms, constituting major sections of scientific study. This allows scientists to aim on a field and can make science much easier and better to apprehend. The study can enlighten and educate people to ask worries, come to conclusions, determine confirmation and declaration, solve issues revolving various forms of life. With the growth of medical and healthcare industry, life science subjects, sales and marketing has become a field of internet among medical students and disciplines completely dedicated to the living organisms study. Life science is an extensive area which includes study related to various fields like biotechnology, bioinformatics, bioengineering, virology, microbiology, bioethics, cell biology, and developmental biology. The single biggest threat to man's continued dominance on this planet is the virus. The Nobel Prize winning biologist Joseph Lederberg once wrote, few epidemiologists would disagree. There is no bomb, no poison, no plan of attack with potential to do as much damage as virus can. Today, the entire mankind is on its knee because of an invisible viral entity, suggesting the significance vigorous research to unravel new insights and unprecedented novel micropathogens that are yet to evolve. This is going to be a continued challenge to sustain life on this earth and in ever changing internal and external environment, further signifying the need of life science research more so ever. We are all aware how technological changes, especially those in the life science, research at an unprecedented scale and pace. On one hand, we see a lot of promise in them for fostering new advancements. On the other hand, we face severe challenges. For example, the quest for the COVID-19 vaccine discovery. The whole world scientific fraternity working together to achieve this goal. And how more opportunities are being created and transparency is growing with online intermediation. New invention and discoveries are taking place on a frequent basis, facilitating human lives. While we reap their benefits, we must prepare to address the challenges too. Being committed to deliver on our goals of preserving the integrity of life science research and protecting the interest of life scientists, we need to be alive to these developments and keep our instruments and toolbox under continuous monitoring and review. Our interventions in the rapidly evolving space in this field must be guided by understanding of the theories of harm, comforting with facts and evidence. In that, sharing experiences will help to stay up to date, design better strategies, and optimize enforcement and advocacy efforts. Though this webinar, we are trying to play an instrumental role in facilitating useful and relevant knowledge sharing. Respected delegates and dear participants, this webinar will present a well-published knowledge sharing by few internationally acclaimed scientists and will create awareness on the recent trends in life science research. I have no hesitation in saying that the webinar will reveal facts which were not known to us or will propagate the knowledge. We shall present and discuss the findings in detail the technical session. We expect that this will be springboard for exchange of ideas on how to uncover and deal current challenges in this field of research. With these words, I once again welcome you all. The technical session ahead have been well thought of. We compliment the steering group of our head of the Department of Zoology, Dr. Pragya, planning and putting together topical issues for brainstorming in an international platform. I hope that this webinar will be yet another opportunity to do lessons, to draw lessons and best practices from one another, which can take back. Thank you all, and I wish this webinar a great success. Thank you. Morahela. Next.
Good morning, everyone. Now is the time. We will begin our technical session with the speeches of our delegates. Before starting the technical session, I request all the participants to kindly attend all the speeches by the delegates and raise their questions in the text box after the end of the talk. Now I am going to invite our first speaker, Dr. Santosh Kumar Pandar, who has completed his master's in Ravensa University and achieved his PhD in biotechnology from Utkal University. He has Dr. Panda has 14 peer-reviewed journals and has reviewed 20 papers from different journals. I invite Dr. Santosh Kumar Panda from School of Medicine, Washington University to kindly present his talk on the topic Coronavirus, a challenge to immunologists. Dr. Panda, please kindly start your talk.
Hello everybody, respected principal sir and other eminent faculty members, and thanks Swagatika for this nice introduction. Am I audible to everybody? I want to just confirm.
Yes, sir. You are audible.
Okay. And could you all see my slides also? Anybody could reply just.
Yes. Your screen sharing is means it is
Okay.
we can view your screen.
Okay. So today my topic is coronavirus, a challenge to the immunologist. As all of you know that the contribution of immunologists to this world is mostly other than other applications, the best contribution to the world is vaccines. So vaccine is the medicine which was really discovered way back 1600, but till date every day it's a challenge for the immunologist to find new vaccines, new vaccine candidates, safer vaccine candidates, and as well as mostly effective vaccine candidates. But when this coronavirus pandemic came in, how it becomes a challenge to the immunologist, I'm going to talk about these things. So before going to detail into the topic, I just want to introduce what is coronavirus. So as I know that there are audiences from different background, I just go to the basic of it. This coronavirus is actually not new. This is a myth that this is a new virus. This is not a new virus. Rather, it was discovered way back in 2000, I think 2003 to 2002, in that late 2002. That virus was a mild virus whose effect is not that not at this scale. But this virus, which appears now, it's a mutated strain of coronavirus. That's why we call it as coronavirus 2. And as it poses a severe acute respiratory syndrome like SARS virus, that's why we renamed it as SARS-CoV-2. And this disease was actually, the pandemic was started in Wuhan, China, from the pneumonia patients in late November of 2009. That's how we are calling it as COVID-19. As all of you know, the global pandemic causing death of around 17 lakhs of people. And now every day it is counting. I think you all know, if you check the World COVID Meter data website, at every second somebody is dying out of this virus. And I think you all see how the laborers in our Indian streets, they walk miles and miles. People got jobless. It has not only biological impact, it has much more social impact as well. So let's see how coronavirus actually affect our life. It is not going. So sorry for the interruption. Actually, the computer get hanged. So the symptoms of coronavirus are, if somebody is affected with coronavirus, first there are what are the symptoms of coronavirus? There is a fever. It have chills, including rigorous fatigue, cough, sore throat, shortness of breath and breathing difficulty, loss of smell and taste. Symptoms start about 1 day after exposure, but it can be as long as 14 days. That's why there is that 14-day you have to be isolated if somebody by chance affected or by chance exposed to somebody with coronavirus without a mask. That's why there is a 14-day isolation. But there are evidences now, recent evidences saying that up to 21 days somebody could be a carrier without any symptom. And some people have no symptom, most have a mild illness, but if you have additional complications, it can be severe and sometimes fatal. This is a graphics of spread of coronavirus. You could see the red, which are deep red, like India, Russia, United States of America, Brazil, Argentina. There are much more incidences and much more death. Whereas those who are with the faded color, they have less incidence of coronavirus and less death. So definitely, as I am talking from USA and my home state in India, everybody is affected through coronavirus severely. So let's have a look into the structure of the coronavirus. It's actually, as all of you know, virus is nothing but a nucleoprotein particle. Nucleoprotein means it have a nucleic acid core as well as a protein envelope. So this is the protein envelope where it is a lipid membrane, and there are some envelope proteins which otherwise are these membrane proteins which act as protease inhibitors. The most prominent part is the spike proteins. The spike proteins act as antigen and act as the infectivity of this coronavirus based on these spike proteins. And this is the RNA which actually the core of this virus and having the infective gene. Next, let's see how replication of this virus takes place. So in past, as all of you know, there is a door to entry. Likely in cell biology or in immune system, this is called as the receptor. These are called as the receptors. These receptors like SCE2, TMPR, SS2, these two receptors are were initially identified as the receptor of coronavirus, which recognize the spike protein and let the virus into our nasal epithelial cells. But in a recent science paper, people also identified neuropilin. Neuropilin is another receptor for coronavirus. But let's look into these two. When coronavirus get into our nasal epithelial cells, then it uncovered, the RNA comes in, and then it use our host protein synthesis machinery. By using our ribosomes, it synthesize many more proteins and replicate the RNA. Then it get enveloped, and lakhs of virus released from a single cell, which affect other cells as well. So for coronavirus detection, we use RT-PCR method, when the technician, the clinicians take the nasal epithelium, take the nasal epithelium. So there are expectedly there are either there are virus or there are no virus. If there are virus, they will purify RNA from these cells, and using primer they will do RT-PCR test. If the RT-PCR test is positive, somebody, the donor is or the sample is positive for coronavirus. If the RT-PCR test is negative, the donor is negative for coronavirus. This is the most trusted test, and all over the world now it is, everybody is believing that this is the best test. But there are other tests too, like serological test. Serological test means in this test we used to have antibodies. We test the antibodies. As all of you know, when the spike protein is recognized by macrophage, I will describe it in my in some of my future slides, but it was presented by dendritic cells or macrophages. to the CD4 T cells. Generally, it is go to the CD8 T cells. Then it induce the B cells to produce IgG and IgM, the antibodies. First initial phase is IgM and the next stage is IgG. So we test IgG against the spike proteins. If there is a presence of IgG in the plasma sample or serum sample of the patient, then we call it as he is as corona infected. If it is not present, we call it as corona, not infected by corona. But this test although this is quite easy test than the RT-PCR test, it have quite drawbacks. A recent study identified about 35% of common people having antibody against coronavirus, because the antibodies are cross-reactive in nature. So the specificity of the test is yet to be evaluated properly. Now I just want all of you to have a look what are the key phases of disease progression. First, SARS-CoV-2, this interacts with the spike protein through, as I mentioned, ACE2, TMPRSS2 or neuropilin. Then it entered into the nasal epithelial cells. This stage we called it as pre-symptomatic or asymptomatic stage. So at this stage, although there are no symptoms of the disease, no exhibition of the disease, still he could affect, or he or she could affect some other peoples. Then this virus infect the ACE2 expressing type 2 alveolar epithelial cells, and the patient exhibit pneumonitis. And at this stage, the immune system comes in. T cells, macrophages, NK cells, plasmacytoid dendritic cells, all have to play a role here. Then the severe disease case comes in. So when, as I think you all know, if there is inflammation, if there is encounter to virus, there will be inflammation, which is generally type 1 inflammation with inflammatory cytokines. And these inflammatory cytokines have febrile temperature, and as well as other diseases, means other symptoms, lungs failure, shortness of breathness, means generally they will collapse the lung, as a result of which it will be very difficult to breathe and survive. So these are the immune cells. I want to show what are the immune cells which contribute to the coronavirus. First, if the coronavirus in the lung and some other virus in the gut, these are the pictures showing it. We have fever, dry cough, dyspnea and diarrhea. At this case, I want to highlight these are the PDCs. PDCs are plasmacytoid dendritic cells. These are in other words called as professional interferon alpha producer. As I think we all know, I want to revive that interferon alpha is the most prominent antiviral protein. So the virus, when the virus RNA comes in contact with the PDC express TLR7, which recognize the RNA, and then it produces a lot of interferon alpha, which kills the virus infected cells, as well as activated the B cells and macrophages. The macrophages directly recognize ACE2, could engulf it, could degrade it. The B cells could produce antibodies which could neutralize the coronavirus. As NK cells also have a prominent role to kill the virus infected cells, as well as CD4 T cells, CD8 T cells. Every cells have a role to play to combat the coronavirus infection. This is a simple diagram which shows how the signaling really takes place. I will go through it. There is a TMPRSS or ACE2 receptor which interact with the virus. It comes to the endocytosed, and in the endosome there are, as I mentioned, there are TLR7 and TLR8 which recognize the single strand RNA. This single strand RNA, the TLR7 work with MYD88. It's a downstream signaling molecule which ultimately activate NF-kappa B and IRF7. It is called interferon regulatory factor 7. The IRF7 ultimately produce lot more inflammatory cytokines and interferons. Although inflammatory cytokines and interferons are required for successive combat against the virus, but excess inflammation causes the disease, which is now most scientists, most clinicians implicated that excessive inflammation causes the disease. Here, the same what I told, this is a graphical representation. So the macrophages, naive T cells, in help of the naive T cells and macrophages, they interact as a result of cytokines produced. Then B cells activated. B cells produce antibodies which actually act against the coronavirus to neutralize the virus. Then, as I mentioned in my first sentence, now everybody is waiting one-eyedly for the vaccine. So let's understand how the vaccine works. So vaccine means memory. The word actually vaccine comes from the word bhaka. I think you all read in your basic immunology books. So vaccine, in other words, is the memory response. We have to induce the memory response in our body. So, if you all remembered, means I think you all know, definitely remembered, the polio virus vaccine is nothing but a live attenuated virus. We injected a live attenuated virus to our body, as a result of which our body system get activated. Like suppose there are some firing in the border. It may be mock firing, but our forces get activated, and if a real force comes in, or a real intruders come in, then our force could combat easily. The same way the vaccines work. So when we inject the live attenuated virus, it may be live attenuated virus or antigen, any antigen which induce immune response. So the macrophages and dendritic cells, they will first take the antigen and then they will present it to the disease. DC will present it to the CD4 T cells. The activated CD4 T cells will induce or will activate the B cells, which will be plasma cells, and some B cells will remain as memory cell. The plasma cells, it will produce many more. You understand, it is 2000 antibody molecules per second it could produce, an activated plasma cell. So it will produce antibodies like bullets. So the antibodies will go directly, it will bind to the antigen and it will neutralize the virus. But most important is the memory. If there is a memory inside our body system, our immune system will activate it immediately when it encounter the virus again. So that's how the vaccine works. So the vaccine, if there is a vaccine, it may be, there are different kinds of vaccine: RNA vaccine, DNA vaccine, protein, live attenuated virus, whatever it is. Important is whether how much it could induce memory response. The stronger the memory response, the better the efficacy of the vaccine. So everybody, somebody is raising hand. Yes, Banshidhar sir, I could ask me. Okay. So let's understand how vaccine works. First, there is an exploratory or preclinical stage. So at this stage, the scientists test different molecules, basically mouse. Why mouse? Mouse is easy to handle. It's not that easy to handle a human being, or it is not that easy to handle a dog. Rather, mouse is cheap, easy to handle, and many more mouse could be in a small space. So first, after get exploratory and preclinical stage, which takes generally 2 to 3 years, now then it comes to the phase 1 clinical stage, where we maximum 8 to 10 people we recruit, we inject them and check the efficacy. Then means this is safety, as if somebody is not dying out of it. Then phase 2, it is 100 plus peoples, where this is safety as well as immunogenicity, whether there is memory or not, whether it have any side effect or not, whether it is inducing properly memory response or not. Everything will be tested in phase 2. Once it is past the phase 2, then we have phase 3, where 1000 plus peoples. and then regulatory review. So generally vaccine development is a 10 to 15-year process. Whereas we now at this pandemic, we are expecting a vaccine in as soon as possible in even few months. So this is a really challenge for the immunologists and the clinicians which are working for these vaccines. So this slide, I want to walk through what are the possible targets, how could we combat SARS-CoV-2 or coronavirus through different strategies. First, I already repeated you many times, the SCE2 and TMPRSS2. These are the doors through which coronavirus enters into the cell. So if we shut the door, like we shut the border, like we shut our door, nobody could enter inside our home. Similarly, we have to shut these receptors. To shut these receptors, we have to develop neutralizing antibodies. So if there is a neutralizing antibodies against SCE2 or TMPRSS2, then definitely the virus could not get the doors to get into our system. Other strategy is soluble SCE2. As you could see, the receptor have a transmembrane domain. This transmembrane domain is helpful to induce signaling by means of which it could get internalized. Whereas if you inject soluble SCE2, it could bind to the spike proteins and they could not get in. Then this virus need to be endocytosed. That's how it could release. So like chloroquine, hydroxychloroquine, TMPRSS2 inhibitor, they could not release from the endolysosome, late lysosome. That's how we could block the virus replication. And the other like nucleotide analogs and others we could put to inhibit the RNA replication of the virus. That's how we could block the virus. But although there are many more vaccine candidates, I am not going through it due to limited time. Just I want to explain you the most successful vaccine candidate, which is now already approved by Federal Drug Administration of United States, and now they already started injecting the vaccine to the emergency and healthcare professionals. So this is a RNA virus. So what we did, the scientists did, they identify the sequence of the RNA which code for the spike protein. So they isolate the RNA, and this is a synthetic RNA. Actually, they synthesize this RNA and they packed it inside a lipid coat. So when we inject this vaccine to somebody, to the individual, the lipid coat will fuse with the cell membrane, as a result of which the RNA will be released. The RNA will utilize our host synthesis machinery and will synthesize the viral proteins, particularly the spike proteins. So the spike proteins, the cells, as you know, if anything foreign to our cell, it will present through major histocompatibility complex 1, MHC1, to the or if it will be come to the circulation, the dendritic cells should take it and it will present through MHC2. So that's how there is a presentation of antigens through dendritic cells. And once the antigen presented, as I mentioned in my previous slide, the B cells will come in, they will develop antibodies against these spike proteins, and that's how the immune system will work, as well as memory response will come in. So before ending my talk, I just want to tell all of you what are the challenges immunologists all over the world facing for this coronavirus. I mentioned repeatedly, SCE2 is the interferon stimulated gene which help for the coronavirus to come in. Whereas this table shows in the SCE2 induction in mouse, generally we loved the mouse very much because we could easily do many more experiments in mouse, but the mouse do not express SCE2, which is the receptor for coronavirus. Human cell line, then next what we do, it's easy to test in human cell lines, but you could see interferon gamma production in human cell lines is very less. Whereas primary human cells, it's much more, and it's very difficult to do research on primary human cells. So that's how it's much more challenging for the immunologists, for the clinicians to have a research on coronavirus. Luckily, in our Washington University, an investigator, a very good virologist, they developed a mouse model which expressed human ACE2. That's how they identified some candidate molecules. I think now what I know, they are in a process to develop a nasal vaccine which they could inject directly to the nostril, and that's how they could protect. The second challenge is, as the most successful vaccine is RNA vaccine, you all know RNA is the most unstable molecule in the world. So when we do RNA work, all my research friends and as well as you people must know that RNA is very unstable. With a little bit of mishandling, RNA will be degraded. This is a picture which showing how the United States authorities planning to send RNA in minus 70 degrees centigrade. In India, it will be very difficult for us to train the technicians, which really the ground workers, which will really vaccinate the peoples, as well as to maintain minus 70 with that much of infrastructure and facilities. Not only India, all developing countries will face the same problem. So these are the challenges. And before finishing my talk, I want to say thank you all. Thank you for having me here. And please feel free to ask me any question you have. And stay safe, wear a mask, as well as maintain a social distance. Thank you all. Okay.
ସେ ଲୋକ ଅନମ୍ୟୁଟ କରୁନି ନିଜେ ଅନମ୍ୟୁଟ ହେଇପାରିବ ଏଇଟାରୁ କୋ ଡାଇରେକ୍ଟ ଫୋନ
Thank you, sir, for your informative talk on this topic, coronavirus. Now I request all the participants to ask questions. Any questions from the participants? Kindly put your questions in the chat box. Sorry for the inconvenience.
Okay.
Kavita, kindly write your question in the chat box.
That's okay. Instead of typing, if they could ask directly.
Hello.
Yeah.
Good morning, sir.
Yeah.
Sir, I just want to ask, sir, before asking question, thank you very much for. Sir, my question is that if the scientists develop nasal vaccine, then what will be the procedure of that and the mechanism of that, sir?
Could you repeat your question, Kavita?
Sir, if scientists develop the nasal vaccine, then what will be the mechanism of that vaccine and which protein or which type of mechanism will be there? We can target which protein or.
It's a good question. Thank you for asking this. Actually, the nasal vaccine, as I mentioned, if there will be nasal vaccine, it will directly go into our nostril, and there it will immediately activate the immune cells, because we have some tissue resident. You know, all these mucosal cells. Actually, mucus, in other words, called as second brain, where gut mucus, and this gut mucus as well as nasal mucus have so many immune cells. That's why. So these immune cells will get activated. There are B cells, there are T cells, there are other macrophages. That's how it will directly, the local immune system will directly combat the virus there. That's the real mechanism is against this.
Okay, so the next question is from Dr. Vibhuti Barik.
Yeah.
The question is, what were some of the reported side effects of the Pfizer's COVID-19 vaccine?
It's a difficult question. Actually, we are also wondering. Means as a researcher in this field, every day we are searching for that. You see, there are 1,000 people every day is, either affected with some or some other diseases. So whether this is due to the coronavirus vaccine or it have their preclinical conditions. Now, in this short span, it's very difficult to say whether it have some side effect or it's not that it is safe, because in general RNA, as the research says, RNA could not go directly into DNA and it could integrate the DNA nucleus, host nucleus. So the side effect, it's very hard to say. Means not cell, it's very hard to say. We are not sure whether this is side effect or they have preclinical conditions.
Okay, the next question is from Dr. Gitanjali Raut. She is asking about some preventive measures to the immuno-challenged persons for coronavirus.
It's really a difficult question. Thanks, Dr. Gitanjali Raut. Means right now we don't have any such kind of, means in one word there is no line. I could not say advise anything that the immuno-challenged people could do anything. The better is they have to maintain a distance. They have to avoid the virus. Now, right now we could not say anything about these things. Wear a mask, avoid the gathering, stay safe, stay in a safe distance. That's the best now. Right now, that's the best option.
Thank you, sir, for answering the questions. Due to the time limitation, now we are moving for the talk, and I request Dr. Pragya Paramita Naik to introduce the next speaker.
Thank you so much, everybody. Thanks for having me here.
Good morning, all. The keynote presentation of today's webinar is to be presented by Dr. Sujit Pudhari, sir. Dr. Sujit Pujari sir is presently working as the Director of Viral Vector Core and Assistant Research Professor, Department of Pharmacology and Physiology and Neuroscience from School of Medicine, University of South Carolina, USA. He has also worked as Assistant Research Professor in Department of Entomology, Penn State University, USA, and he has done his postdoctoral in research in entomology, Penn State University. He has 45 publications from highly acclaimed peer-reviewed journals and with 457 Google Scholar citation, and he has published in very reputed journals like Molecular Medicine, Nature Communication, Journal of Medical Virology. He has also received many prestigious awards like winner of Lightning Talk at 10th Annual Postdoc Exhibition PSU USA, Audience Choice Award Lightning Talk at 10th Annual Postdoc Exhibition PSU USA, Office of Postdoctoral Affairs Penn State University Travel Award, Young Scientist Award by Indian Virology Society of India, Gold Medal from Defense Research Development Establishment Government of India for outstanding research work published in Journal of Molecular Diagnosis. Now, I would like to say that we are very much fortunate to have Dr. Sujit Pujari sir among us to deliver a talk regarding the coronavirus, which is the major threat in today's world. We are all aware about it more or less, and we have also faced very difficult situation in today's time because of this coronavirus. So I would like to request Dr. Sujit Pujari sir to be kind enough to deliver his talk and enlighten us with his experience and knowledge. Thank you, sir.
Thank you, Paramita, for the nice introduction. Let me share my screen first. Here we go. Can you see my screen? And am I audible?
Yes, sir, it is audible and we are able to see your slides.
Okay, perfect. Good morning to the audience in India and good evening to the other speakers and audience around the globe. Just to give you an update, the current time at Columbia, South Carolina is around 11:21 of Sunday. Currently it is raining outside and the temperature is 7 degrees Celsius. With that, many thanks to the organizing committee for hosting this webinar. My sincere regards and thanks to Dr. Gopal Haldar, the principal of Bikram Dev College, this event's patron. I also want to convey my special thanks to Dr. Pragya Paramita Naik for contacting me to deliver this talk on SARS coronavirus 2. Also, many thanks to Ms. Swagatika Panigrai for moderating the session. The topic for my today's talk is SARS coronavirus 2, the pandemic of the 21st century. The things we have learned about this virus in the past 1 year. There is some technical glitch here. Give me a second. Okay, here we go. Since we have a very diverse audience for today's talk, I would like to keep my presentation as simple as possible. With a goal of our presentation will be to brief you some of the fundamental concept in infectious diseases. Also tell you more about SARS coronavirus 2 and the current pandemic. Also, we will discuss about the vaccine development process in general and the status of SARS coronavirus 2 vaccine. For my talk, we will start with an introduction about infectious disease and pandemics. That will follow a brief discussion about SARS coronavirus 2, its origin, how the virus entered into the cell, and what are the clinical symptoms, also about the course of clinical illness in the patients. Also, we will discuss about the different diagnostic methods, which can tell you whether you are currently infected with a virus or SARS coronavirus 2, or you were previously infected with that virus through antibody testing. That will follow the prevention part, where we talk about the vaccine in general and also current development in SARS coronavirus 2 vaccine. At the end, we will conclude with a question answer session. So without any further delay, let's be to the topic with a very simple and fundamental question. What is an infectious disease? Well, an infectious disease is a disease caused by microorganism that can invade tissue. It could be a virus, a parasite, or a bacteria, or a fungi, or even a yeast. And these microorganisms are transmitted by contaminated water, sometimes air, contaminated food, or bite of or an infected animal or an insect. If we see or analyze the number of outbreaks that happen or caused by the microorganisms in last few decades, what we see here is that the virus and bacteria has basically take the lead over fungi, parasite, and protozoans. And more importantly, if you see in last decade or so, the viruses are continuously threatening the human civilization through continuous outbreak and epidemic or pandemic-like situation. Yes, you heard the right word, like the pandemic, outbreak, epidemic. These are the words basically what as an infectious disease we use to describe an infectious disease or how it basically occur in a population. To give you an idea, to let you know what it is, so what you see here is like 3 squares, and the area inside the square is basically representing the area of a particular geographical location, and each yellow dots represent an infected individual. So if you see the box to your left, where a particular disease occurs like randomly in a particular geographical location, then in that case we call that is a sporadic occurrence of the disease. When it appears in clusters, that's what we call that as an outbreak of a disease. But if it is random, like it's spread all over that particular geographical location, that's what we call an epidemic. But when a disease goes global, we call it as a pandemic, right? So let's do a little bit of time travel. Let's go back to early part of 20th century. In 1918, the humankind see the one of the biggest pandemic, what is called Spanish flu, which was the causative agent was influenza virus, which wiped out almost 1/3 of the world population. Let's fast forward to again 1957. We got Asian flu. Again, it was influenza virus, which killed almost 1.1 million people around the globe. In 1968, we had the Hong Kong flu virus, which killed almost 1 million people. HIV, the killer virus, which appeared in 1980 and still is around us, in last 40 years, it has killed almost 35 million people. And it is still killing more and more people. In 2009, we saw the pandemic of swine flu virus, which killed about 757,000 people around the globe. And still it is there, and it is now here and there popping out as a normal seasonal flu. And now in 2020, we are experiencing the pandemic of SARS coronavirus 2. Wait, did I miss something? Okay, let's zoom into the last 20 years or so. What was happening in last 20 years? Yes, there are again a bunch of viruses there, which created a pandemic, not really pandemic, but pandemic-like situation, also threatening the human population with severe outbreaks. Let's understand why so many pandemics in last 20 years. So in order to answer this question, we need to understand what were those infections or what were the origin of that virus. Let's see one by one. In 2003, we had the outbreak of SARS coronavirus 2 or SARS coronavirus 1. The origin of that virus was bat. In 2009, we had swine flu or H1N1 pandemic. The origin of that virus was pig. In 2013, it was MERS. The origin of that virus was camel. Again, in 2016, we heard a lot about Zika virus. The Zika virus is basically, again, it stay in a forest, in a sylvatic cycle, or in a forest cycle. They have like the mosquito, the monkey are the reservoir, and they are happily like basically circulate in the forest between some of the monkeys and other wild animal and mosquitoes. And in 2020, we are now experiencing the SARS coronavirus 2. We do not know the exact, from where it originated, but what we know so far is that the closest relative of SARS coronavirus 2 is in bat. So what is the like common thing you see in this slide? Most of these viruses, like in fact, all the viruses, they have an origin, they were originated from an animal. And when a particular disease originated from animal and come to human, those are called zoonotic virus, or those kind of things are called zoonosis. So how basically the zoonotic disease are transmitted to human? Let's understand the mechanism behind that. So what happened is like, sometime when we come in contact with an infected animal, like it could be in a farmhouse, or in when you come contact with your infected pet, or sometime when you go for a picnic or go for hiking or biking. During that time, sometime what happened, we get exposed to some of the viral infection through those infected animals. At that time, that particular event is called spillover. And from there, two things can happen. One, because since this virus is coming from another animal, it need to adapt into your body or into a new host. That is, the new host is the human now. So if it can adapt, then you will have a disease. If it cannot adapt, then that will be an abortive infection where nothing will happen there. Again, if something happened, like the first thing is happened, like where the virus get adapted to your body, and then you will get the disease. And during that process of disease, the viruses keep replicating, and there is the viruses keep trying to evolve and try to adapt to the human body. And if it's like very well enough in adapting in the adaptation process, the second thing can happen is it can infect more people. That means now the human to human infection is established. If that happens, there's the dangerous point. At that point, that particular virus acquire a character that is has of pandemic potential. So now the question is, which virus can cause a pandemic? Since we have already experienced so many pandemic in the past, with that experience, what we have learned is that a virus, or basically which is a very new strain of virus that, or basically let me say, rephrase it again. So basically what happened, like if a particular virus has a certain characteristic, like what I have listed here, then it can cause a pandemic. The first one, the virus has to be a new strain or a very new virus. Why it has to be a new virus or new strain? Because that virus has not been exposed, or the current population do not have any kind of immunity to that virus or to that pathogen. That means the whole population is now susceptible to that particular new strain of virus. The second, the virus has to be like RNA virus. More like, just forget about the pox virus. Those are the only group of virus, the DNA virus that can cause pandemic. But mostly, most of the pandemics are so far that we have experienced are from an RNA virus, because this group of viruses, they do not have a basically proofreading activity during the replication process, and they do have a very high rate of mutation. The third is the high infectivity. That means it can spread very quickly. You must have heard about the R value, like R naught value of the SARS coronavirus have a R naught value of more than 2, more than 3. Those kind of things you have heard. That's R naught value is nothing but is the how rapidly the virus can spread from one person to another person, and how many person it is affecting basically. The low mortality is also a very important part, because the virus has to sustain. Sorry, am I still connected?
Yes, sir.
Give me a minute, please. Okay, so the low mortality is also very important because the virus need to sustain the infectivity, and also the long incubation period is like a deadly combination for the pandemic viruses, because if in that case the person is infected but he do not know, and they keep spreading the virus in a population. So but basically now, if a particular pathogen is like satisfying all these criteria in addition to the few other criteria, then it can cause a pandemic. So the next important question is like, who decide and declare a pandemic? Is the World Health Organization, or WHO. The World Health Organization is continuously monitoring the different infectious disease. They have collaboration with different laboratory at different part of the world, and they are collecting data about infectious disease like in a very every week, or in some case every day. So whenever they see any pathogen which basically fulfill all the criteria what we discussed in the previous slide, and few other criteria what we have not discussed here because of time constant, at that point basically the WHO declare a pandemic. Anybody in the audience remember this date, March 21, except those who do not have their birthday on March 21st? Anyway, this is the date when WHO declared the pandemic for SARS coronavirus 2. And as you know, like this virus was originated in Wuhan, a city in China. So what happened, it was like basically in December 8, there was like almost 41 cases of respiratory illness surfaced. And then in January 7, they were successful in isolating a virus, and they identified that the virus to be a novel coronavirus in January 8. And in the meantime, they were like did the genome sequencing of the virus. And on January 10th, they basically released the sequence and published the sequence in a public database. So now let's see like whether the SARS coronavirus 2, I'm sorry, I wrote it SARS coronavirus 1, it has to be SARS coronavirus 2. Whether this SARS coronavirus 2, does it satisfy all the criteria what we discussed previously? Is it a new strain of virus? Yes, it is a new strain of virus. This is a RNA virus. It is very highly infective, but it is spread very quickly. It has low mortality in order to sustain the infectivity, and it has a long incubation period. So basically, it has all the deadly combination with it to be a pandemic virus. So when we analyze basically the genome sequence of the SARS coronavirus 2, what is available now in the public database, what we found out that this is a coronavirus 2, like SARS coronavirus is belong to the family Coronaviridae and fall under the genus coronavirus. So basically the coronavirus has like again 4 different subgroups, alpha, beta, and gamma. And the SARS coronavirus 2 falls under like a beta coronavirus genus. This is not the first time the humankind is seeing the coronaviruses. Before that, we have already witnessed 6 different coronaviruses. The 4 of them cause a very mild symptom. You can just get a mild flu-like symptom, mild cold, and then automatically it goes away. But in 2003, for the first time, we see a very severe form of the coronavirus in the form of SARS coronavirus 1, which has a mortality rate of almost 10%. But surprisingly, it suddenly disappeared in 2005 or 2006, and after that, it never came back. Then thereafter, in 2013, we saw another coronavirus, which is called like MERS coronavirus, which is more like one of the deadliest among all the coronavirus, where almost 30% of people die if they get infected with MERS coronavirus. And now, we are experiencing one of the biggest or like a pandemic of coronavirus, which is SARS coronavirus 2. Luckily, it is not very fatal. Only 1 to 2% of people die because of SARS coronavirus 2. Again, this is the structure of of SARS coronavirus 2. It is a envelope virus. It has a positive sense RNA genome, which is like protected with a envelope of lipid envelope. And the lipid envelope is basically, decorated with some protein called spike protein, which basically it gives a crown-like structure, and that's why you get its name coronavirus. And then all of you know this is an airborne infection like other coronavirus. And since all of you know, whenever you go outside, that's one of the reasons, because it's an airborne virus, you put a mask whenever you go in public or when there are many people around you. I will not go more detail into that, but what I would like to tell you is that, like, whenever somebody gets infected with SARS coronavirus 2, three things can happen. One, either you are get an asymptomatic infection and nothing will happen, or you can get a mild infection with just might be a fever, cough, and you are done. Or the third is the more concerning things, where you get a severe infection which can end up with a multi-organ failure, or the patient may die. Why does this happen? Why is this discrepancy? Like, why some people are asymptomatic? Why some people are get the severe infection? The reason, possible reason, is that sometime if suppose you are fall in the group of asymptomatic individual, that means your immune system is very, very good in identifying the SARS coronavirus 2. It kicks in time and remove that viral particle from your body. That's how you are asymptomatic and you do not have any problem. The middle one is the mild infection. In that case, your immune system is good, but it's get activating little bit late. But anyway, even if it is late, it works. That's how you get the mild infection. But in case of severe infection, sometimes what happens, in case of some of the individual, their immune system could not identify the virus in time, and when it identify that virus, then it get confused and what to do, and then it immediately starting producing different immunoregulatory molecules that cause cytokines, antibody that recruit more cells to kill the virus. And while doing so, it forget about or identifying the cell and versus non-cell. That means what is the pathogen and what is your the own body cells. And during that process, it start killing your healthy cells, and that's the reason the patients get the multi-organ failure, and sometimes they die. So what makes the SARS coronavirus 2 more infectious than the other coronaviruses? So in order to answer this question, or in order to understand this, we need to know some things, some important things during the viral infection. The first thing, is the virus should be able to attach and enter the host cell. That's the very important thing. The second is it should be able to efficiently replicate inside the cell. The third, it should be able to modulate the host immune system. That means it should be able to basically trick the immune system and escape the host immune surveillance system. So these are the three things a virus need to have this characteristic in order to infect and cause a severe infection. So let's see what happens in case of coronavirus. So this is basically I'll show you a model how the virus enter into the cell. So this is the last spike protein present on the cell's virus surface. It has two domains, S1 and S2, decorated with glycan. When it comes in close to the cell membrane, basically the receptor identify that, and with the help of certain proteases, it removes the S1 domain. Basically, that helps to unzipping and folding and refolding of the S2 domain. And after and during this event, basically, it's pulled down the viral particle close to the cell membrane, and that's how the virus fuse to the host cell membrane and release the viral particle into the host cell. So once the virus is the viral genome is released into the cell, then the replication process starts. So we'll discuss that later, but let's first see the structure of that S protein, because that's the important protein which allow the virus to enter into the cell. So if we compare the S protein of the coronavirus, which cause mild disease, what you see in your lab, and the structure of the S protein, which cause severe disease, what you see in your right, there is a difference there. You can see the picture in your left has a close conformation, whereas the S protein on your left, they have a partial open conformation. That means the partial open conformation means that means they can easily enter into the cell, because that's how it expose some of the critical residues, so that the the host protease cleave those protein and allow the release of the S1 protein, so that S2 protein can bring the virus close to it and allow for the fusion process. So again, but the question is still there, like what makes SARS coronavirus 2 more infectious than all the other coronavirus? So what we have seen in the previous diagram is that those coronavirus which cause severe infection, they do have a partially open conformation. But if you see again the S protein, the sequence of S protein of SARS coronavirus 2, here what you can see that it has acquired 4 different amino acids during the process of evolution. And while doing so, because of that 4 insertion there, it also acquired a furin cleavage site very next to the S2 protein. The furin are basically a serine proteases found in both cell membrane and inside the cell, which basically accelerate process of the cleavage of S1, so that the S1 or the red part go away, and so that S2 protein can pull down the virus and allow the virus to fuse with the cell membrane. And notice the third thing is again, this virus can also utilize an additional molecule called neurophilin-1 or NRP1 as an additional receptor molecule. So basically now this particular virus, SARS coronavirus 2, use both ACE2 and as well as NRP1 as the receptor, so that more and more virus can enter into the cell and very efficiently. So in summary, what we can say that SARS-CoV-2 virus evolved with an efficient entry mechanism, which make them more infectious. So once it enter into the cell, the second thing is to it need to replicate. So it is a very efficient replicator there. Then once replication is done, then it produces the other viral protein, and ultimately it again those viral protein are assembled and produce infectious viral particle, which are released outside of the infected cell. Either this infected cell could be in your nostril or could be at other part of your body. Again, keep answering that question, what makes SARS coronavirus 2 more infectious than coronavirus? Again, we need to let's see this picture. What you are seeing here is the replication pattern of the human coronavirus, what we know so far. The picture in your left, which is in little bit blue color, this is for the coronavirus, which cause very mild symptom, like mild flu-like symptom or mild cold. They basically replicate in your upper respiratory tract. Whereas what you see in your extreme right, which is a little bit in green color, those kind of virus or that include the MERS coronavirus and the SARS coronavirus 1, they replicate in the lungs and they cause more severe from the disease. But again, if you see the SARS coronavirus 2, they can replicate both at the upper respiratory tract as well as lower respiratory tract. That makes them more they are severe, they can produce severe disease, as well as they are an efficient transmitter, because they are there in their nostril and producing more and more infectious viral particle. So whenever you are breathing or you are talking, the more virus are getting expelled to the outer environment. Again, when after somebody get the virus, the first 2 weeks are very critical, and this is the time you may get a fever, cough, or some mild mild symptoms. And at that time, after 2 weeks, the person may get clear the virus and they are perfectly fine. But most of the whatever happen is at that particular border point. The other what can happen is a group of the other people who get into the severe form of the disease, they start experiencing hypoxia condition may appear. They need to be hospitalized and they need to put in the ICU. And during the whole process, your body oxygen level goes down. That result in a basically the blood clotting activation factor goes up. That may result in kidney failure or acute cardiac injury. And also like sometime, many secondary infection happens and ultimately the patient die. So basically what is the summary is that the first the second week is the more crucial point in a SARS coronavirus 2 infection. So also you have seen that or you have heard that in many of the time that the person who got infected with SARS coronavirus 2, they lose their basically the sense of smell and taste. This is because when the virus is replicated in your upper respiratory tract, basically it replicate in the nerve ending which are there in the olfactory bulb area. And also during that application process, it also use it as the port of entry into the central nervous system. Again, this virus is like very kind of tricky and very notorious. That means even if it is like a respiratory virus, it can get access to like different part of your body, including your central nervous system. And there are many reports where some patient with SARS coronavirus 2 develop neurological symptoms. Again, as I told you, this is primarily a respiratory virus. That means it attack your respiratory system. So many of you who are many of you must know or might know that lungs is the primary organ where the blood exchange happen. When you breathe, the oxygen is taken up by your body and it is expelled the carbon dioxide, right? So the alveoli are basically the structural and functional unit of lungs, where basically these are some balloon-like structure which are covered with with your basically with blood capillaries. So where deoxygenated blood comes, and because when you breathe, the oxygen exchange happens and the oxygenated blood goes other way. So in case of SARS coronavirus 2, what happens, the lining of what it covers the alveoli, it get inflamed. And because of the inflammation and also, the body try to, fight that virus in the lungs. During that process, it release or basically it sends some of the neutrophil macrophages into that particular area. And during that process, the lining or the endothelium lose its, the integrity. And because of that, the fluid accumulate inside the alveoli. And because of the accumulation of water there or the fluid, now the blood exchange is severely hampered. And because of that, the patient goes into a hypoxic condition. Because of the hypoxia, again, your blood clotting cascade is activated, and when the blood clots start moving in your body, that can cause severe life-threatening problems. Again, in case of in general in any viral infection, the disease severity can be attributed to the viral factor or the virus, where sometimes what happens in the viral, the number of viral goes significantly high, so that your body cannot handle them. Or the second thing that attributed the disease severity is the misfiring of the host immune response. The immune system get confused, and instead of sending an appropriate signal, it start sending some weird or inappropriate signal for that particular infection, and that's one of the reasons when people get the severe infection. Or the third scenario is where the both, because of the increase in the viral load as well as in the misfiring of the immune response, can cause disease severity. So let's see what happens in case of SARS coronavirus 2. So does viral load lead to a severe disease? So in order to understand this question, the scientists around the globe are analyzing more and more data, analyzing or studying the more clinical samples. And then still there is no consensus there. There are some people say yes, when the viral titer goes up, the patient go into a more severe form of the disease. But there are some data where they could not find any correlation between the viral load and disease severity. That means we need to have more data in order to answer this question. Again, in order to answer the role of immune system, first let me give you in a brief what are the cells present in the blood. So as you know, in our blood we have red blood cells. It has plasma cells, it has platelet, And also it has, like, more importantly, the white blood cells. The white blood cells are basically army of your body. These are the immune cells which basically identify the any pathogen or any invader that enter into your body, and they identify them and kill them. So this can be, like, classified as, broadly, like lymphocyte, eosinophil, basophil, neutrophils, and monocyte. And each group has their own role, and they get activated in different kind of situation. In case of a viral infection, basically, most of the time the lymphocyte come forward and they try to fight with the viral infection. And while doing so, like, they basically activate two different kind of immunity. Like, one is called like the innate immunity, and another is called the adaptive immunity. So when your body is get infected, the innate immunity immediately come to play. The innate immunity, basically, what this arm of immune system does, it do not recognize what is, it's a, like, virus, or like a bacteria or a pathogen. It just look for something that's not self, then it start killing those. In the meantime, while the innate immunity is doing its work, the adaptive immunity, which are like more specialized kind of thing, what they do is basically identify, or whether it is like bacteria or it's a virus, and they try to identify what are the, like, the protein present on the cell surface of that particular pathogen, or like what are the, like, some of the other key factor which cause the disease. So they identify them, and they try to develop or try to produce certain kind of antibody or some kind of cell, so that they can fight that particular infection. So in summary, what we can say, like, the innate immunity is a non-specific immunity which can randomly identify cell versus non-cell, but the adaptive immunity is your, like, very specific, and it just specifically attack those virus, and those cells are infected with that virus or bacteria. So this data is from a study where they have analyzed the different the change in the blood cell type during the SARS coronavirus 2 infection. What you are seeing here is the, like, B cell, T cell, whatever the other immune cell in the percentage in a healthy human being, or the healthy control. But when you see in a SARS coronavirus 2 infection, what they found, that there is a significant decrease in the T cell and dendritic cell, which is a not common thing, especially for a viral infection. Also, they seen an increase in the monocyte and increase in neutrophil and eosinophil. Again, this is not normal. So that means there is something weird thing is happening because of the SARS coronavirus 2. So that the person who get the severe form of infectious disease or the SARS coronavirus 2 disease, they cannot fight the infection properly. Again, you must have heard, like, or you must seen in the news that there is a new strain of coronavirus in UK. There is also, I saw, or you must have noted that there is another new strain of virus in South Africa. Is that true? Is the SARS coronavirus 2 mutating? The answer is yes, because this is a RNA virus, and it do not have a proofreading activity, so that it can, during the process of replication, it can rectify those errors. It cannot rectify that because it do not have that capacity. That's why it's accumulating those mutations. And you see, like, what you are seeing here at the bottom corner, this is the Wuhan strain, and now what you are seeing here, these are the strain emerged from that during the last 1 year in different part of the globe, and they are mutating at a rate of 22 basically substitution per year. But does this mutation increase virus infectivity or disease severity? Frankly speaking, we do not know yet. But what we have seen so far is that there is a variant called D164Z, which has replaced the original Wuhan strain in many parts of the globe. But does this variant has any role in the disease infectivity? People have done some study. They have seen that at least in cell culture model, this particular variant is highly infective. But in human, we do not know it. We do not see any kind of such signal from that particular strain of virus so far. The next question you may ask, and is there any impact of that mutation on the vaccine efficacy or the effectiveness? No data yet, but any possibility again? if you ask me again, then I will say yes, there is a possibility, because this mutation is occurring everywhere in the genome, but the chances are very, very low, because in order to be to have any impact on the mutation rate, the mutation has to be at the site where the virus neutralizing antibody binds. So at this point, I'm not worried about that, because that's nature of that RNA virus will keep mutating, but this virus is not mutating at the alarming rate, so that we should be worried about. So the next question you must be very interesting, interested to know when this pandemic will end. The question is whenever we'll have a herd immunity, as simple as that. The answer is simple, but is in practical is not that simple. How do we get a herd immunity? What is that herd immunity means? The herd immunity means basically your body already have seen that a particular pathogen and you have an antibody to that, or you can have a kind of memory to that virus. And for that particular SARS-CoV-2, we do not have any antibody to it, because our body has not seen those kind of viral strain before, right? So in order to answer that, let's see let's see what happens. Suppose a new virus come, a person get infected, because none of them have any antibody against that virus in that population. That means that entire population is highly susceptible to that virus, and within no time that the whole population get infected with that virus. And this is exactly what happened with SARS-CoV-2. So, but in case of vaccination, what happens is basically, without getting, all the population get infected with virus and get the antibody, because during that process, many people die, and we do not want that. And that's why we need a vaccination, right? So during the process of vaccination, we vaccinate the people, and whenever the vaccination, reach a point of around 60 to 70%, when the 60 to 70% people have antibody against that virus, and that's the point when this virus cannot, spread very effectively, because there are not enough susceptible people around infected people. So that's how basically we can stop a pandemic. So as I told you, during that process of vaccination or infection process, your body produce a lot of antibody, and because of the increase in the antibody, you do not get the infection, or you fight the infection, or you can clear the infection. So basically, how this antibody works, let me explain you that. As I told you that once the virus is inside your body, it basically your body immune system, basically specifically the adaptive immune system, scan the whole virus and see what are the different kind of protein on the viral cell surface. So basically, it take fragment of all those, keep a very detailed analysis of that, and then it start producing antibody against those viral protein. So whatever it can produce, let's say 10,000 different kind of protein, and out of these 10,000 protein, say only 5,000 can bind that virus. But again, from that 5,000 different kind of antibody, only few percentage of protein, when they bind to that virus, basically they can inactivate the virus. So that's the very important group of antibody, and these group of antibody are called neutralizing antibody. And this neutralizing antibody basically clear the virus from your body. And basically, this brings to the concept of again vaccine. What vaccine does, the vaccine basically train your immune system to produce those kind of antibody which can inactivate the virus. And during that process, your vaccination process, after 2 weeks of vaccination generally, or sometime 3 weeks, your body start producing more and more antibody. And then if you get infected, then immediately your body clear the virus from your body, and you're perfectly fine. So as I promised you, I will tell you about how vaccine is prepared. Basically it has three main steps. One is the first is the research step, where you do the understand the virus. Then the second step is the testing, and the third is the manufacturing step. So let's go one step at a time. So in the first step, or during the research, what we do as a scientist or as a virologist is first we analyze the virus, what kind of virus it is, what are the different kind of protein it express on the cell surface, because those are the protein very important, because that's how your body see the virus for the first time, like the virus, how it look like, and also how your body is responding to that virus. That is, understanding that part is also very important, like whether it is activating the innate immunity first, or activating the adaptive immunity. Then again, in adaptive immunity there are different type of cytokines or chemokines or antibody they produce. So once we understand that, like how the body is interacting, how immune system is interacting, and how the virus look like, so from all this data we pick few vaccine candidate. And this step is, believe me, is very, very important. If this stage is not done properly, then you might end up with a wrong vaccine candidate, and then your vaccine development things will fail at some point of time, or some testing point. So we do not want any compromise in any of this step. On the next step is like the testing, where we test the vaccine candidate in human population in order to test their safety, efficacy, and the side effects because of that virus. And so once the testing is done, then we basically the company or whoever is delivering designing the vaccine, deliver that formula to a company. Those company basically manufacture the virus in a very, very large quantity, so that we can deliver that vaccine and vaccinate more and more people in less time point. Again, in an ideal condition or in a normal circumstances, the research, testing, and manufacturing, all these taken together, it might take 10 to 15 years. But in a pandemic, we do not have that much of time, because people are dying, people are suffering. We need to come up with a solution in a very less amount of time. And during that process, basically, we squeeze out many things without compromising. Again, I'm repeating this, without compromising any of this, any of them, like testing or in the research, so that we come up with a very safe vaccine. So before going further, I would like to tell what are the different kind of vaccine that are available, or like how we make a vaccine and what are the different type of vaccine. The first one I would like to tell you is about is the attenuated vaccine. This is the one type of vaccine that produce a very strong immune response, but making this virus take a lot of this vaccine take a lot of time, because what we do here is we passage this, like we grow this virus in a different host for a long period of time, until this particular virus lose the power of creating disease in you. Again, it will be like a live virus. It can infect you, but it will not produce any kind of disease in your body. So that's why, it takes a very long time. Again, producing in a large quantity of this particular type of virus is very difficult, and it's time consuming. The next one is the inactivated vaccine. So in this case, what we do is we grow a lot of virus, and we take them and irradiate them with the UV ray, gamma ray, or some kind of radiation, so that all the virus are dead. Or what we can do, we can treat them with some kind of acid, some kind of base, so that the virus is dead. So and then we purify them, and we can use this as a vaccine candidate. But again, this vaccine is not very good, or like it cannot produce a very good immune response, because this is dead. There are a lot of change have been happened because of that, irradiation or during the process of inactivation. And again, production of this is kind of, you can produce more, but again, it takes time. The next is the protein subunit vaccine. In protein subunit vaccine, what we do, as I told you, during the process of research, we again, analyze the different viral protein, and during that process, we identify that which against which particular protein our immune response is making antibody. So that's what we find out. And which we use that particular candidate as to make our protein subunit vaccine. Again, this is not as efficient as the live attenuated vaccine. Another group of viral vaccine is the viral vector vaccine, where what we do is we work with different kind of vaccine, like viruses that infect, like say chimpanzee, or cow or pig, that cannot infect human. That means if you inject the kind of virus in a human body, then they cannot cause any they will not cause any disease in your system. So we basically identify those viruses and develop them as a vaccine candidate. Then what we do is, we basically remove some of the part of that animal virus or the viral vector, and then we incorporate that particular gene of that viral protein of interest, and then we make a new recombinant virus, and we use that virus as a candidate, as a vaccine candidate. And here comes the hero of the story, the mRNA vaccine. So basically, these are like a very new technology what has developed a few years back. If I remember correctly, it was developed for making virus for flaviviruses, for West Nile virus. So what it is done here is basically, again, here we identify the exact protein against which our immune system produces the neutralizing antibody, and we take the gene from that virus and make mRNA of that, and then we encapsulate those mRNA in a basically nanoparticles, and then we deliver those mRNA into the human as a vaccine candidate. And this is so far we do not have anyone before SARS-CoV-2. We didn't have any mRNA vaccine candidate that has been approved, but recently we got the approval for the first mRNA vaccine approved for the SARS-CoV-2. So what we discussed, what I tell you is there are different kind of vaccine that can be made, but with their own advantages and disadvantages. But again, what I tell you, the first experimental vaccine we got is as early as 10 days or within 7 months for Zika virus. But again, we were able to break that record, and we got the first experimental vaccine for SARS-CoV within 42 days of the first the genome get released for this virus. And again, having that experimental vaccine is not well enough to have a vaccine, because the next step is the testing, and this is the step which takes the longest time, because we need to be damn sure that this particular vaccine is safe enough so that it cannot produce any side effect, and it can have a efficient. It should be effective enough to reduce the disease in an infected person. So during this process, as I told you, we have phase 1, phase 2, and phase 3 trial, and we do that in different groups, and it takes time. But in a pandemic situation, we do not have that much of time, because again, I told you, the people are suffering, people are dying, and we want to stop that. So here we become, scientists become a little bit more innovative. So what we do is, first we check whether is the toxicity of that particular vaccine candidate, or what we call the phase 1 testing. Once we have some preliminary data from the phase 1 testing that we know that this is not toxic, then at this point we start the phase 2 and phase 3 trial in parallel, in order to save time, because we already know that this is not toxic. People are not dying out of that. So now the next step is to identify which actual the dose is well enough to have that can produce a very the efficacy of to understand which can produce a very effective vaccine candidate that can protect you from a viral infection. So in order to know that, we do the phase 2 and phase 3 clinical trial in parallel. And then once this is done, the next step is to bring those data to a competent authority like FDA in United States, or similar authority in other countries, where they basically analyze each and every of the data and they analyze it very critically, so that they do not miss anything, or this virus is entirely safe for the population. So once they clear it, basically it goes to the manufacturing unit for and then ultimately come to the market. So for SARS-CoV-2, there are many groups who are working. In the earlier part, we had more than 200 vaccine candidates that were being tested. But we are very lucky, and we are in the humankind for the first time that a vaccine has been generated within 8 months. That's in a record time. And right now we have two vaccine that has been already been approved by FDA and European agency. And out of those, again, those vaccine candidate, one vaccine candidate that was, I think, possibly, if I'm not wrong, that is from like in Australia, they were developing and it failed the safety issues and they have been abandoned in the midway. And then again, we have like 6 more vaccine that have been, approved for limited use. And there are many other vaccine candidates. Those are on the pipeline, which has come to the market sometime sooner or later. Again, those 2 viral vaccine, what I told you, those are one from the Pfizer, which is the big vaccine company, and the second one is the Moderna vaccine candidate. The another is developed by Moderna, which is a company based in United States. So this is how their vaccine candidate work. I told you, like, the spike protein is on the surface of the virus, and this is the very important protein which the first immune system see that particular protein. So and we know that all the neutralizing antibody are against this basically the S protein. So what scientists have done is they see they analyze the genome of that virus, which basically encode for this S protein, and they took this the S protein the sequence of the S protein and make a lot of mRNA of that, and then they encapsulate the mRNA of that S protein in a nanoparticle and use that for vaccination. So once a person gets vaccinated, what happens is that the mRNA enter into a cell where enter into the cell, and where basically it produces the S protein from that mRNA, and this S protein are expelled out to the out of the cell, which is basically again recognized by the host immune response, or immune basically and the body produce antibody against this protein. And once basically when you get infected with the virus, then immediately this antibody come into rescue, and they kill the SARS virus in your body and protect you from further any infection or complication. And in the Indian perspective, basically right now the Pfizer and AstraZeneca, another company which is working with Oxford, they have already submitted document to the Indian authority for licensing for that vaccine in India. I hope it will be like done quickly, and India will see the vaccine soon in the market. And again, India also working on some indigenous vaccine candidate. Those are like Bharat Biotech has Covaxin, and Serum Institute of India Covishield, and also there is another candidate called Covovax. So these are in the very end stage of testing, and hoping this will be done soon and will be in market in next couple of months. So while all these things are happening, it will take some time, but it's very important to protect yourself and protect your loved one through a social vaccination. Isn't it like cool, like social vaccination, a cool word? This is nothing like a fancy. It is like it's a very simple. You need to protect yourself by keep washing your hands, following the basic rules or what you already know. And the second one is not infecting other by protecting, having a mask. So this is the time where we have to take the responsibility of our own, and also the responsibility to protect our neighbor or protect our loved one. So basically with this, I will end this presentation. And if you have any question, I would be happy to take them. Thank you very much.
Thank you so much, sir, for this detailed explanation on the topic, starting from the structure of the virus to the vaccine, from sporadic to pandemic. It was really an informative session. Now, I request the participants to text their questions in the chat box.
Okay, I think it will be easy for me if like somebody, if they ask me question directly to me.
Yes, sir. Yes, sir. Any questions you can directly ask to the delegate. Okay, sir. So here we have a question like which sequence of the RNA in coronavirus makes it more prolific?
Prolific means what? In what sense? Like, can you be like little bit more specific? That?
It was by A. Murmu from Rairangpur. I think she is asking like which sequence is responsible for the particular infection.
So, okay. So I didn't get her his question.
Yes.
Like what I'm saying, let me explain to him is that basically what happens, like any viral infection, the very first important thing is to the virus need able to like enter into the cell. For that, like the SARS coronavirus 2, the S protein is the protein responsible for like entering into the cell. The important thing is that this particular RNA virus or the SARS coronavirus 2 has a like kind of like open conformation, like a half open conformation. So that because it is, remember my slide, I told you that SARS coronavirus, which cause, basically, normal flu or the normal cold, they have a close of the S protein. But the SARS coronavirus 2 has a partially open. And if you remember, you have seen that particular animation, it is very important for the virus, the S1 part, S1 sub removed in order to diffuse. So if because this SARS-CoV-2 has a partially open conformation, the protease which cleave the S1, they get access easily and they can enter the cells very quickly. If that answered that question for him.
Okay, so moving to the next question, Jyoti Prakash Rath has asked the question, as both Moderna and Pfizer vaccine is mRNA based, so why the efficacy varies?
Yes, that's a very interesting question because there are some proprietary things they have not disclosed. Again, as you know, like mRNA is not very stable and it can be easily destroyed once it is inside the cell. So what I believe is that like one of them, they have designed that mRNA such that it can stay inside the cell for a long time. That means they must have protected some kind of cap at the 5 days and 3 days end of the mRNA, which is like in one for one company it might be like working very efficiently and the other one might not working efficiently. That's what I can see. And also there might be what kind of polymer they are using for the encapsulation of the mRNA. So these are the like two very important factors that can governing the efficacy of that virus, because ultimately both of them are mRNA for the S protein, and those are the protein which is producing the S protein against which the antibodies are being made.
The next question is from Himanshu Charan Sahu. He is asking, if I got an infection and subsequently got recovered from it, should I still need a vaccination?
So yeah, the current recommendation is that you need to wait for at least for a couple of weeks after the infection, because if you are infected, you should not take a vaccine, and even after you have a infection, then you should wait at least couple of weeks you need before taking a vaccine. So your question, or the audience question was like, one, I have a infection, then shall I get my vaccine or not, right? So as you know, like currently what data suggests, when you get an infection, the current the antibody against that virus lasts for roughly for 3 months or so. That's what the current data says. So I would recommend to take the vaccine. Currently we do not have any data for our vaccine, but I believe that the current the antibody level or protection given by the vaccine is more than 6 months. So yes, he should take the vaccine. Okay, I can see somebody is like writing that, is it mandatory for a healthy person to take the vaccine?
Yes, sir.
So it is not mandatory, but at the same time, it's a like social responsibility to take the vaccine, because being a healthy person, being a like suppose if you are in your 20s or 30s, you will not be like greatly affected because you are not in the risk group. But if you do not take the vaccine, then if you get infected, it's very high chance that you can you can transmit that infection to your loved one, elder one who are at the very and a risky group. So in order to protect your society, protect your loved one, you need to take the social responsibility and take the vaccine. And again, everyone should know that right now, whatever the vaccine we have, there is no no data that is saying that once you take the vaccine, you will not transmit the virus. Whatever we are saying, the efficacy means, like in the current sense, the efficacy means once you take the vaccine, you will not get the severe form of the infection for sure. So that means you are protecting yourself. But at the same time, you are maybe keep shedding the virus. That means you are still you will infect more virus. So even if you take the vaccine, it is very important at this present time point to protect yourself and other by having a mask, and also keep following the normal routine of washing hands and those kind of things.
Sir, would you like to take any more questions?
Yes, sir.
Yes, so there is another question the vaccines developed from the Western countries, will it be work for the Indian strain?
So, well, this virus is, particularly is mutating, but it's not mutating at a fast rate. So basically, what I can say is, it should be efficacy. The vaccine should be effective enough in human, the Indian population too. But again, it's up to the the authority who will take the decision, either whether they need any breeze, some kind of experiment where they want to do some kind of vaccine efficacy trial in human population. It's up to them, because again, when now if you see the entire globe is multicultural, the different the people, even if in United States there are many people from India, they stay here, and there are many Indian they could be there in that vaccine trial. I do not know that, but I believe there are many Indians who must have taken that vaccine. So we can gather those data, and from that data that can be interpreted, because we are again coming from in the Asian lineage, or Indian, our ethnicity is from Asia. So if it work here from we Indian who live in outside the country, then it should work in India too.
Thank you, sir. So the next question is, after recovery from COVID infection once, can the virus infect the same person twice?
Yeah, there are a few reports where people have demonstrated, or the literature is demonstrating that yes, you can get the second infection. Because as I told you, if you get a very mild infection, then your immune system may not produce sufficient neutralizing antibody, or the memory T cell or memory B cell there. And then again the antibody level drop very quickly within if you get a very mild infection within 1 month. So there is again you can get infected. The short answer is yes.
Okay. Thank you, sir. So another question is, how far the genome sequence of the COVID different from COVID, SARS-CoV? I think he's particularly asking about the sequence of different virus strains.
Yeah, it's quite different, and very important part is that there is a difference in the S protein. You saw that in the S protein there is a 4 insertion in the junction between S1 and S2. So there is a difference in both SARS coronavirus 1 and SARS coronavirus 2.
Yes, sir. So another question is, what is the effect of climate change on the vaccine globally?
Well, its effect on vaccine, I do not see much correlation, how the vaccine and the climate. No, I can see there is, because
Actually,
of
what is the virus? He's trying to ask that what is the impact of climate change on virus, the
yeah,
infection?
yeah. Obviously, that there is a big, you can see because of the change in climate, there are many things are happening. There is the temperature is going up, and there are many, suppose if you give a very nice example is the transmission of Zika virus or the mosquito-borne viral infection. There are many mosquito species which used to only stay in the tropical region. Now they are in the temperate region because of the increase in temperature, and that's how those viruses are now present in global in different part of the globe. So yes, the climate has a very significant role in the emergence of all these viral infections.
Sir, probably the last question is why COVID virus shows less T memory cells.
Well, this is like interesting question, like why they have less T memory cell, but till now we do not have much very concrete answer to that. If you see, but anyway, if you see or remember, like one of the slide I showed you, that the SARS coronavirus 2 infection, basically the T cell population decreased dramatically. We need to understand there is a lot of work need to be done why this kind of event is happening. So once we know that basic mechanism behind that, maybe at that time point I'll be like in a good position to answer that question.
Yes, sir, definitely. And another question is like the recovery rate is quite high in India according to the statistics. And Sweta Praharaj is asking that what must be the possible reason to this?
I think first of all, I would like to say like we should be very happy about that, right? The recovery rate
Right.
is.
Yes, sir.
Okay, so let me give you like an example, because like we Indian, like we always like get exposed to certain low dose of infection from outside. Suppose when we eat something street food, or like when we drink water, there is a little bit of high level of microorganisms compared to the like what supposed to be. So because of that, like we are constantly taking some kind of microbes into our body, so our immune system is always active. So might be that one of the reason we are not getting severe infection, or our body is like acting very quickly or fast enough to clear that viral infection.
Thank you, sir, for nicely explaining all the questions. It was really a descriptive session by you, explaining the virus very in detail and answering the questions. Thank you so much, sir. It was really a nice presentation by you. Uh, Now we will move to the next speaker.
Thank you. It was my pleasure. Thank you.
Thank you, sir. So moving to the third speaker of today, Dr. Sanjay Kumar Sahu. Currently, he is a postdoctoral fellow at Pulmonary Rheumatology Department, Washington University School of Medicine, USA. He has achieved his PhD in biotechnology from Bose Institute, Kolkata University. His work focuses majorly on complement biology. He also worked on understanding the regulation of immune response genes in Mycobacterium tuberculosis infected macrophages. Dr. Sahu has published his research in some highly reputed journals like PLOS Pathogens, Cell, Host and Micro, Cellular Microbiology, Scientific Reports, Journal of Biological Chemistry with 442 Google Scholar citation. Now I request Dr. Sanjay Kumar Sahu to kindly start his talk. Dr. Sahu, please.
Yeah, thank you. Good morning all to all of you, and thanks for your nice introduction. Yes, I'm sharing my screen. Mm. So are you able to get my sound properly and the screen share what I am sharing now? Yeah, what I mean. Yeah. Hello everyone. Are you able to get my voice properly and the screen what I am sharing now?
Yes, sir. Yes, sir, the screen is visible. You're audible
And
too.
yeah, and my okay, thank you. Just a minute. Good morning to all of you. I am thankful to Dr. Gopal Holdar, Principal of Bikram Dev Autonomous College, and all the staff members of the college, and Dr. Pragya Parimita Nayak for contacting me to present, to deliver one talk. So, as all of you know, this is a global pandemic is going on due to the SARS-CoV-2 or COVID-19. And I heard the last two talk of Dr. Santosh Panda and Dr. Sujit Pujari. So they talked about the coronavirus and the efficacy of vaccine. So because of this COVID-19 or because of this pandemic, people were more acquainted with the webinar, the virtual talk, virtual seminar. So, but as in my point of view, because this is a imbalance between the nature and the human interference about the natures and the human, we should always balance between the nature so that this type of pandemic it should not happen in future, because as this coronavirus is from a bat, and it is from a in Wuhan, from that meat markets. So anyway, so we must take the responsibility to preserve, to respect both to the environment, to the wild animals and everything, so that this type of thing it should not happen again and again in future. So my talk is basically on complement and immunity, as I am working on complement system, and I am also working on the COVID-19, this is SARS-CoV-2 on the immunological perspective point of view. So just here today I am going to just describe the basic concept of complement and how the complement plays important role in the immune response. So everyone needs a complement now and then. And what is the role of this complement, and how our ancestor and how we are surviving because of these four factors, that is protection, the food supply, the blood, and the bugs. Bugs gives immunity, blood clotting, food supply, and the protection escape. So what is the role of complement? It protects, because complement is basically a group of proteins, serum proteins, consist of more than 50 serum proteins. They are present, they are always in inactive state when the pathogen enters, or any type of factors, foreign materials or any particles enters into the body system. They recognize and they maintain an immune response. So this is a diagrammatic representation of the live bacteria. This is, you can see, this is a killed, and this is the lice. So discovery of complement. So between 1885 to 1900, the heat stable serum factor and heat labile serum substance require, these two factors required to lyse the bacteria, lyse the RBC in a transfusion reactions, lyse RBC in an autoimmune disease. So heat stable serum factor was specific during vaccination or natural exposure, and heat labile serum factor was non-specific. Everybody had it. There is no need of any type of vaccination required. So due to the discovery of serum, the role of serum in the immunohemolysis and the complement protein, Bordet got the Nobel Prize in the year 1920 for the discovery of this complement system. And after that, Louis Pilmer, who discovered propyridine, and what is the role of this propyridine in the alternative pathway of complement activations. And I will discuss about the different complement pathway, how complements different pathway are there, how they recognize. So as you know, there are two parts of immune system. One is antibodies. Antibody means that is specific, acquired. That is the antibody that it is not inborn. This is the acquired immunity. And innate immune response, what is it? That is the complement factors. Innate means inborn. No need to prior exposure to any antigen or anything else. It is inborn. By birth, we have the complement system that is secreted in our cells, from the hepatic cells, from the monocytes, from macrophage, from the adipose tissue. So first autoimmune disease detected in the year 1895, that is hemorrhagic anemia. And the blood transfusion reactions required antibody to lysis the RBC blood cells. So complement biology, the history in 1970s, alternative pathway was rediscovered. In 1980, complement receptors and what is the role of this complement in the inflammation and opsonizations. In 1990, there is the lectin pathway. In the year 2000, the crystal structure of C3. And in the year 2010, the intercellular complement system. This is that earlier thought was that the complement protein is secreted only from the hepatic cells of the liver, not within the cells. But now that thought has changed, and within the cells, monocytes, macrophage, some cells within the cells, they also secrete some complement proteins that is called intercellular complement system. And what is the role of this complement in the different genetic disease, and how we apply these complement proteins for the drug discovery or to cure any disease. General concept of complements. So this innate immunity, as I mentioned, innate means the inborn immunity. the that complement protein it circulates in the blood in a inactive form, that is called as the jamogen, and they are when they bind to the any type of bacteria or viral, they got activated by cleaving. This is serine protease just cleave and they turn to active forms. So how the complement system works? The complement system, the complement proteins that binds to the antibody, antigen and antibody. It just like a in between and helps in the opsonization. The process is called opsonization. Mean molecule that binds to the both antibody and to the antigen and helps in the phagocytosis of the that virus or bacteria, any other foreign particles. Then when the binding takes place, the activation takes place, serine proteases. Then there is the interaction between the thyroid bond and the covalent that is interactions. Then the amplification takes place, and finally the attack, means the membrane attack complex C5b9, the final outcome that rupture the virus or bacterial cell wall and lies the virus particles and the bacteria, all the foreign particles, particularly the microorganisms. So how the complement system works? This is as you can. This is the membrane modifications, the pathogen, opsonizations, lysis. This is anaphylatoxins. Anaphylatoxin means it when the C3A, C5A, these are the two molecules of complement proteins that plays important role in the inflammations. Inflammation means where there is high level of IL-6, TNF-alpha, IL-1 beta, and that leads to the release of migration of neutrophils, monocytes, and if there is uncontrolled inflammation, that leads to the death, or otherwise called septic shock. The complement protein plays important role in the the C3A, C5A in anaphylatoxins. There are three different types of pathway by which the complement work. One is the classical pathway, another is the lectin, and the third one is the alternative pathway. The classical pathway, where the antigen binds to a particular antibody binds to a specific antigen on pathogen surface. On the pathogen surface, the C1q, particularly in the classical pathway, the C1q binds to the pathogen, and where to the C1q the antibody binds and it helps in the clearing out of the pathogen or the phagocytosis of the pathogen takes place. In the lectin pathway, manoj binding lectins MBL that is present on the surface of the in the cell wall of the different pathogens, bacteria. So that particular MBL when binds to the pathogens, it activates the complement system and generates and the clearing out of the foreign particles that is involve the lectin pathway. And most prominent pathway, that is the alternative pathway, where the C3 pathogens, where the C3 molecules binds to the pathogen, and after binding to the pathogen, the C3 molecules cleave into C3A and C3B, and the factor B will come that binds to the C3B molecules and helps in the lysis of the pathogen through membrane attack complex C5b9. So three pathways of complement activation, the classical, lectin, and alternative pathway. But the C3b, it is the most important one, C3b. It results in the inflammation, opsonizations, and in the lysis through C5b9 complex. So this, when the C3b molecule binds to the pathogen, any other foreign particle in virus or bacteria, whatever it may be, the foreign particles, immediately the opsonization of antibody response takes place. And it helps to recognize the antibody, so that antibody will recognize well to the pathogen and helps in the clearing out of the pathogen. And
नहीं इनके तरह तो नहीं, वो तो पहला वाला था नहीं, that does not determine कि तुम less.
This is the another is the C3, that as I mentioned, the serine protease C3b and C3a. Here different opsonization, inflammation, amplification, and membranated complex, that is the C5b9 that takes place. Complement activations. This is the target cell. T means target cell. C3 is cleaved by converted C3b. Here the factor B, factor D, and factor P. Factor B helps in the cleavage of, factor B binds to the C3b, that is cleaved by factor D, and P the propyridine, that it stabilize the factor B. So this alternate pathway, then the C3b also activates the positive feedback loop taking place. So in this way, the alternate pathway, the proliferation, the complement activation, and the amplification also takes place. And here, that is the same, the target molecule, the C3b molecule binds. This is the alternate pathway that going on. The C3b, BB, and P, this is the convertage. It cleaves the C3 molecule into C3b and C3a. C3 and C5a is the anaphylatoxins. It helps in, means it, when there is increased level of C3a and C5a in the cells, means there is high level of inflammation. And what we have seen here in, as in VASU, some COVID patients, we are analyzing the serum samples or plasma sample of the COVID patients. And what we have seen that in those patients who have died due to this COVID-19 have high level of factor D in their serum and high level of C3A and C5A. Means high level of inflammation taking place. If the expression of C3 and C5A is high, it leads to the increase in the expression of pro-inflammatory cytokines. The three pathway of complement activation, as I have mentioned earlier also. This is the classical lectin and alternative pathway. This is the inflammation lysis also. The reason why I am putting the picture of a snake, the cobra. This is the cobra venom factor. It also this is a non-toxic protein from the cobra venom. That also, when you treat any human beings or any animals, any human beings, that can this cobra venom factor also induce the alternative pathway, the C3b complex, C3b pathway. The another pathway, this is the complement activation of lectin pathway, and then this is the lectin pathway, and is a classical pathway. The C3 molecules, this is a target cell, and C3b binds to the target cell. And because all the pathway, lectin pathway, classical pathway, and the alternative pathway, all three pathways converge together at one point, that is the generation of C3b molecules. That C3b molecules helps in the inflammation, opsonization, lysis, and clearing out of the pathogens. This is manoj-associated serine protease, that is C1q, that is a classical pathway, C1q, C1 receptor. Here in classical pathway, C4 and C2 this C4B, C2A, another convertage that also cleave the C4 molecule, and then helps in the lysis of the C3 to C3B and C3A. And how the complement works, means the C3 molecule binds to the pathogen, then the activation of serine protease takes place, then membrane engaging, then through this membrane the target cells, this C5B through 9, this is the final outcome of the C3B, the complement activation pathway. It lies the membrane of the bacterial wall or the any pathogen molecule. So how the complement proteins binds, because different receptors are there for different complement proteins, but so the complement proteins bind to the different receptors and they perform their different functions depending upon the stimulus. So complement system and immunity. So the complement system acts as a first line of defense, as it is a innate immune response, and helps in the innate immune response, not the adaptive one. Innate immune response in helps in the clearing out of the pathogen, and it helps in the to complete the reactions, means a thing that completes or brings to perfection. So antibody cannot recognize the antigen without the presence of this complement alone. So complement helps in the opsonization also, so that antibody can act properly to the pathogen. Means antigen identification, processing, cellular activation, co-stimulator molecules, cytokines, and also humoral immunity. Then complement therapeutics, different C1 in hereditary angioedema, C1 inhibitors, kallikrein is an inhibitor of factor D, gain of functions or loss of functions, monoclonal antibody to C5A due to the means to check the prevention of septic shock or the excessive inflammations. And recently this eculizumab this medicine has been given to some COVID patients to check the inflammations, and they have tried to another 10 to 20 patients. I didn't remember exactly how many patients they have given this drug, but some of them survived in the ICU and some of them died. They have tried to, means after authorization, they have tried to use this to check the excessive inflammations. So complement role in Gibbon disease. So as it is a innate plays a role in innate immune response circulates in the blood in an inactive form. It protects the bacterial infections from viruses, bacteria, autoimmunity preventing cellular damage, chronic inflammatory disease, helps to clear out the tumor cells, binds to the tumor cells, and after binding to the tumor cells, the antibody recognize that whether this is the foreign particles or not. Then the antibody mediator then targets to that tumor cells and helps in the clearing out of the tumors. And drugs in the clinic. So this is the end of my slide, and thanks for your patience hearing, and I would like to address any questions from your side.
Thank you so much, sir, for your presentation. And now I request the participants to write their questions in the chat box.
Yeah, they can directly
So
ask me the questions, no problem. Yeah.
Yes, sir. So the first question is from Dr. J.P. Ratha, and the question is whether septicemia is due to overreaction of complement system.
Yeah. Yeah, septicemia is or excessive in the that is called this is mainly due to the C5A and C3A, that is they mediate the that is called anaphylatoxins, and due to the excessive inflammations, I mean due to the secretion of these two proteins, means the activation of this complement pathway that leads to the release of C3A and C5A, and that C3A and C5A leads that increase in the production of IL-6, TNF alpha, and that in turn results in the uncontrolled inflammation, and that leads to the septic shock.
Okay, sir. The next question is from again Dr. J.P. Ratha, and he's asking for the mechanism of action of danazole.
Yeah, danazole that it blocks the factor B. It blocks factor B so that factor B cannot be cleaved. So if factor B cannot be cleaved, it cannot associate with the C3B molecules. If C3B is not associated, the convertage will not form. So the reaction will stop. So it check the alternative pathway function, complement activations.
Okay, so the next question is again he's asking that is there any specific reason for COVID, the induction of COVID and the higher response of complement system? Like he's trying to know the connection between COVID and the complement system.
Yeah, it is a good question. Yeah, recently just we submitted one paper in JCI. So what we have means analyzed or we have seen that in COVID patients, the release of mitochondrial DNA is very high. And the mitochondrial DNA somehow it increased the complement pathway. And we did some analysis and we found that due to this mitochondrial DNA binds helps in the complement activations, and that leads to the more production of C3A and C5A, and that leads to the septic shock and death. Maybe due to the viral infections, as you know, the mitochondrial DNA is released from the cells and it may activates the complement system inside the host cells.
The next question is from Padmaja Desai. Sorry, the question is from Dr. J.P. Ratha, and he's asking that whether complement system is present in plants or not.
It is a very great question, but I have no answer to that, because whether the plant system has any complement system or not, because nowadays people are working on the also plant microbe interactions. I think, in my opinion, I would say there must be some, not the complement system, maybe the how the plant resists themselves from the pathogens. Because of them, there must be some type of interaction between the host and pathogen. So there must maybe on some sort of complement type pathway available, exist in the plant system.
Okay, thank you so much, sir, for answering all the questions with a nice effort. And so we are, if there is no question, we will move to the next speaker. Thank you so much, sir. Your presentation was very informative and nice, and enlightening us with the role of complement system in immunity, and giving an addition to its role in COVID infection. Thank you so much, sir.
Thank you.
Good morning.
Good morning, all of you. Our next speaker is Dr. Sujit Kumar Bhutia. I would like to give you a brief introduction about him. Dr. Sujit Kumar Bhutia is presently working as Associate Professor, Department of Life Science, National Institute of Technology, Rourkela. He has done his PhD from Indian Institute of Technology, Kharagpur in cancer biology and done his postdoctoral research in Virginia Commonwealth University, USA in same field, that is cancer biology. Dr. Bhutia is an eminent autophagy scientist from India. His group is focusing to understand the role of autophagy and autophagy-dependent cell death in cancer for development of novel therapeutics. They are identifying the crucial factors governing the crosstalk between autophagy and apoptosis to describe the mechanism controlling cell survival and cell death. He has published 110 articles in various peer-reviewed national and international journals like Autophagy, Apoptosis, Trends in Biotechnology, Hepatology, Cancer Research, Advances in Cancer Research, PNAS, Seminars in Cell and Developmental Biology, Oral Oncology, International Journal of Cancer, and many more. He has also co-authored an article with 9,417 citations. His Google Scholar citation is 14,165. He is also the academic editor of PLOS ONE and reviewer of many internationally acclaimed journals. He is also recipient the various prestigious awards like Professor A.R. Rao Memorial Young Scientist Award CRCA in 2018 and is also recipient the Odisha Vigyan Academy Young Scientist Award in 2015. He is also received ICMR International Fellowship for Young Indian Biomedical Scientists and Young Scientist Award 2015 and ICMR International Fellowship for Young Indian Biomedical Scientists in 2015 and 16. We are very much fortunate to find Dr. Bhutia among us to enlighten us or spread the horizon of our knowledge in the field of autophagy. Now I would invite Dr. Bhutia to kindly deliver his talk and give a very brainstorming session regarding the topic.
Thank you, Pragya. Thank you, Pragya, for so nice introductions. I am fortunate to present our work on autophagy in cancer, the friend and foe. So autophagy is a highly conserved process starting from yeast to mammals. And autophagy have many basic roles. Is it slides are visible?
Yes, sir, they are visible.
So autophagy start with a clearing process. Auto means self, phagy means eating itself. So it eats the unnecessary material and degrade the unnecessary process. So like we clean our home, so it is the housekeeping clearing system of our cells. So autophagy have many basic roles, and it is protect the cells from starvations, from microbes, and it also involved in the repair mechanisms, as well as it have also act as a programmed cell death type 2. So autophagy is connected with the phagocytosis with the lysosomes. There is formation of autophagosomes with formation of double layer structure around the defective organelles of the cells, and then they are degrades with lysosomes releasing nutrient to the cells to manage the stress. So this is the process of autophagy. Autophagy initiated during stress. So when there is stress, autophagy induces as a protective mechanisms to manage the stress. Autophagy, there is mTOR inactivations. mTOR is a nutrient sensor. So when during stress, mTOR undergo inactivations, then there is activation of autophagy initiations. So ULK complex along with the other proteins, so they activate the autophagy, initiate the autophagy. Then autophagy initiation leads to activation of the phagophores, the double layer structure around the close to the endoplasmic reticulums, where there is activation of VPS34 complex, which is known as a PI3 kinase class 3 complex, which elongates, which produce the PIP4, PIP3, and elongates the membranes, double layers membrane around the defective organelles or defective cytoplasmic materials. Then there is elongation systems which help them the elongation and curvature formations around the cytoplasmic material. Then ultimately it forms the autolysosomes, and autolysosome after that fuse with lysosome for autolysosomes, where degradation takes place and provide the nutrients to manage the stress. Moreover, there is another process that from the autolysosomes, there is formation of lysosome, because this system is continuing. So the system has a developed new process known as autophagic lysosomal reformation, from which the lysosome are formed and continuing this system. So this is the overall process of autophagy. And this is in detail complex mechanism of the autophagy. So autophagy is a cytoprotective mechanism, and it is reversible upon stress removals. So when there is no stress, then autophagy can back to the original conditions. But during prolonged stress, exposure to long stress, so the autophagy is associated, autophagy cell undergo cell death known as autophagy dependent cell death. And autophagy have many functions. They have a role in cell death, they have a role in cancer, they have in immunity, in a cardiovascular system, in aging, and it have a difference in infectious disease. Recently also some scientists are telling that it have a role in the COVID-19 management. So that's why there is some report that chloroquine also can inhibit the COVID infection through regulation of autophagy. So many neurodegenerative disease, maybe there is also say that the like fasting also promotes autophagy and keep the human healthy and decrease the aging effect. Our ancient sages, ancient Muni and sages, so they perform different fasting in Ekadasi and others, yes, they do fasting. Maybe they know that fasting involves in autophagy inductions, and that autophagy induction degrade the cytoplasmic content to keep this cell healthy and human healthy. Also, there is a report that during walking, morning walking also have autophagy inductions and keep the human healthy. So these are the different role of autophagy in different diseases. We are working on the role of autophagy in cancers and how we can manage the autophagy during cancer progressions for a potential cancer therapeutics. We have published many papers. So we found that autophagy have a dual roles in cancers. So like autophagy, as I'm telling, it is involved in the cellular protections as well as it also involved in the cell death. So these are different points where autophagy involved in the involved in cancer, either in pro-tumor, involved in the promoting the cancers, or in pro-death. That's why I have chosen the name that it's a friend or pro in cancer. So I will now explain some work. There are many more literature regarding the role of autophagy, and here I will discuss what our laboratory is working on the autophagy, what we have published and reported. That research I will explain here. So autophagy in and tumor suppressor. Autophagy have a, here I will discuss the tumor suppressor nature. So there are many genes involved in the autophagy, and it's so that many like Beclin-1, ATG5, and many other genes have been deleted during cancer progressions. So indicating they have a tumor suppressor role in cancers. Even it's so that the mice deficient in Beclin-1 are more susceptible to different type of cancers, including lung cancer, hepatocellular carcinoma, and lymphoma. And as we discussed, autophagy act as a cell death mechanism, tumor cell death, program cell death type 2, so which can be used as a approach in for targeting cancer, because cancer cell are the resistant cells, apoptosis resistant cells. Moreover, along with apoptosis, it can act as a complement system to have a two type of cell death in the cancer. So that's why scientists proposing that it have a tumor suppressor role. So we studied different type of genes, autophagy genes in the cancer perspective. We have taken one gene ULK1, which initiates the autophagy, as I indicated in the earlier slides. So we overexpress ULK1 genes and check its apoptotic effects in the cancers. So when we overexpress the genes, and we found that there is strong proliferative effect as indicated through cell MTT assay and colony formations. Next, we checked in presence of anti-cancer drug cisplatins and found that it potentiates the effect of cisplatin in killing the cells and activate the caspase activity, so indicating the ULK1 trigger caspase dependent apoptosis. Next, we investigated what is the mechanism behind it, and then we come to the role of mitochondria, and we found that in the mitochondria there is ULK1 upregulate the reactive oxygen species and decrease the mitochondrial membrane potentials. So coming to the how it induces reactive oxygen species, we found that ULK1 inhibit the superoxide activity, mitochondrial superoxide, that is MNSOD activity in the cancer cells. Next, we found that ULK1 is localized, present in the mitochondria, and it interacts with the MNSOD, leading to inhibition in say activity. So this is the conditions. So in normal condition, there is less ULK, so no cell death. Whereas when you are overexpressing the ULK1, so there is increase in ROS production, and increase in ROS generation leads to inhibition the membrane potential, and ULK1 also internalize to the mitochondria, where it binds with MNSOD and inhibits the activity, and that leads to cell death. We also checked that another autophagy protein, that is ATG14s. ATG14s, which involved in the initiation of autophagy, is also promotes apoptosis in cancer, indicating that these autophagy genes Here we checked ULK1 and ATG14 inhibit cancer cell death through apoptosis induction. Then coming to the protective autophagy, another side of the coin. So we found that autophagy also involved in the tumor progressions. So during cancer progressions, so autophagy, because cancer cells are in stress, nutrients and oxygen stress. So during that conditions, autophagy act as a protective mechanism. It sustains the cell viability in cancer cells, which are defect in apoptosis and long-term metabolic stress conditions. So it provides the nutrients to the cells, basically central area of the tumors, and it's so that many cancers have a high autophagy as compared to the in cancer cells. And autophagy also protect the cells from the therapy. During therapy, so autophagy protects the cells. So in this case, autophagy should be inhibited for potential cancer therapy. So that's why the role of autophagy is context dependent, depend on the situation. So I work during my postdoctoral time work, I work on a gene that astrocyte elevated gene 1, so which highly overexpressed in different type of cancers, and they have a different functions. So I investigated so whether this sort of genes induces autophagy and involve in the protections. So we overexpressed the genes and found that there is increase in the autophagy inductions. Like, so the green puncta, the gene LC3 is involved in the autophagy, and there are many more genes involved in the autophagy, and found that there is increase in LC3 puncta, indicating that there is autophagy inductions. Then we confirm through different approaches. We also confirm through electron microscopy, where we found that enhanced dense double layer structure of autophagism, indicating that there is autophagy inductions in the normal cells. So we checked then again using different approaches. So where there is increase in LC32, it is a lipidated form of LC3, which associated with the double membrane structure of the autophagosomes, and found that in the different normal cells, so there is increase in the autophagy during AG1 overexpression. So then we checked with the knockdowns of the different autophagy genes like ATG5, BECLIN1, BPS34, and found that there is, when we knock down, in ATG5 condition, only the autophagy is decreased, indicating that ATG5 is involved in AG1 dependent autophagy. So what is the role of AG1 in cancers? So we found that AG1 is high in cancer and autophagy also high in cancer. So we knocked down ATG5 in cancer cells and checked the effect toward the stress toward the cancer cells. So when we checked the response to the drug, we found that in the autophagy knockdown conditions, the cancer cells are more sensitive toward the apoptosis, indicating that inhibition of AG1 induced autophagy enhance its to the chemotherapeutic drugs. Next, we have explored another genes. So where that is a clustrin, which also activated in cancers, and we checked whether clustrin induces autophagy and whether these genes have any role in the cancer through autophagy modulations. So clustrin is a chaperone, molecular chaperones, which have a role in different perspective of the cancer. It inhibits apoptosis through BAX, It's activate the survival pathway, PA3 kinase pathway. It increase oxidative stress. It activate NF-kappa pathway, and it also inhibit the cell, protect the cells from anti-cancer therapy. So we checked then expression of overexpression of clusterin and found that there is autophagy inductions in response to. So we assessed using different approach, acrid staining and the LC3 puncta accumulations. So I am happy to tell that this is the Pragyan did all these things during her PhD work, and so we found that she found that there is a co-localizations of clustering and ULK1 inducing autophagy activations. Next, she did with that whether clustering have any roles in the cancer cellular protections through autophagy. So she used different type of autophagy modulations, inhibitors, 3MA and MDP1, which inhibits a particular type of autophagy like mitophagy, and found that when the cells are overexpressed with the clustering and we inhibited autophagy, so cells are sensitive to the stress. So this is checked with in response to serum starvations. Then we checked in response to chemo-resistant chemotherapeutic drug cisplatin. So when we in the AKT in the clustering overexpress knockdown conditions, so the cells are more sensitive to the chemotherapeutic drugs, and autophagy have also same effect, indicating that the clustering induces autophagy involved in the chemoresistance. Recently, this paper we published. And another story coming to the role of autophagy in the cancer. This work also done by Pragya, and we develop resistant cells, oral cancer resistant cells in response to cisplatins, and found that cisplatin resistant cells have a cancer stem cell like properties, and these cells also show high autophagic activity. So she measures different parameters of the autophagy. So where we found that the oral cancer resistance, the oral cancer cells which are resistant to cisplatins have a high autophagy flux, high autophagy status. There is high LC3, 2 proteins, ULK1, Beclin1, ATG5, ATG7, ATG14, P62 decrease, so indicating that cisplatin resistance cancer cells have a high autophagic activity, and it means the oral cancer stem cells have a high autophagy activity. Next, so we found that the autophagy regulates the stemness and chemoresistance in oral cancers. So we in the resistance cancer cells, so we knock down autophagy genes and found that there is decrease in the different markers of the stemness. Like we quantified the beta-catenin expressions, then CD44, these are stem cell markers. So, and indicating that there is correlation between autophagy and stemness in oral cancers. And then we checked the ABC transporters, drug transporter, the different type of genes, and found that there is correlation between the autophagy and stemness in oral cancer. Next, coming to the therapy. So as we discuss that autophagy, role of autophagy in cancer is context dependent, and so it depends on the situation, depend on the conditions. So we found that autophagy in response to therapy, autophagy involved in the tumor suppression, as well as autophagy inhibition also promotes tumor cell death. So two way it have a, so this study, this type of study show that autophagy involves in the cell death and is a tumor suppressor mechanism. So here we inhibited autophagy. The different drugs inhibits autophagy, and there is more when we inhibit autophagy, so there is more cell death, indicating that tumor inhibition, in this case tumor inhibitions have a more effect, whereas here in this case the tumor activations have a more effect, so leading to tumor suppressive mechanisms. So coming to the, we have also studied another interesting finding. So where we found that autophagy have a role in dual role, and so how, which type of when there is protective autophagy or when lethal autophagy. So that's why we have developed a system that so we hypothesis that during initiations, the autophagy is decreased. Then there is increase in autophagy, which protect the cells also. So during cancer progression, during initiation, autophagy decrease. Then after that, autophagy is supporting the cancer for growth. So at the early phase, we call pro-death, and after during progression, we call pro-survival autophagy. This is our hypothesis. So to validate the hypothesis, we have studied the cancer progression in hamster models, studied the progression of oral cancer. It is a chemical induced carcinogenesis, oral cancer carcinogenesis in hamster models. And our study showed that there is normal, this one normal, then hyperplasia, then dysplasia, then neoplasia. This way there is cancer development. After that, we check the autophagy status. So different genes we checked. See, this is in control, normal hamsters, and see, and after 4 weeks, tumor developed till 16 weeks. So after, see, during early phase 4 and 8, autophagy inhibited. And after that, there is again increased autophagy. So validating this hypothesis, this study is going on. And with this, I want to conclude my talks that the role of autophagy in cancer is dual in nature, independent on the context. Thank you all.
Thank you, sir, for your informative presentation. Now we will move to the question answer session. I request all the participants to write their question in the chat box. Is whether there is no copy of ULK1 in genome other than mitochondria.
Can you say again? Please repeat. Hello?
Sorry for the inconvenience. Sir, the question is whether there is no copy of ULK1 in genomes other than mitochondria.
ULK1 is present in ULK genome in the nucleus. So then there is, it can also translocate to mitochondria. It is present in the cytoplasm, but it can translocate to mitochondria.
Okay, sir. The next question is from Dr. JP Ratha again, and he is asking about the role of autophagy in COVID management.
To be frank, I don't know because I don't work in the COVID, but as I have seen in many literature that autophagy have a role in the COVID. So that's why some also telling that they are using the chloroquine have maybe effect, because chloroquine is a fusion inhibitor of autophagism and lysozyme. But detail I don't know.
Thank you, sir. The next question is from Damayanti Majhi. And the question is, how do you differentiate between autophagic cell death and autophagy activation leading to survival?
So there is autophagy, both way there is autophagy inductions. There is both way there is autophagy induction. So we are asking how will difference between whether autophagy is survival or autophagy is cell death. So we have to,
yes, cell death, yes.
We have to see using cell death, different cell death assay, like you can do MTT assay, you can count the cell viability by using trypan blue. When we are treating the drug, whether your cell is going for cell death or cell survival.
Okay, sir. Another question is from Abhishek Mund, and he wants to know what is the role of bioinformatics in autophagy?
Bioinformatics is a tool. Bioinformatics tools. So you
yes, sir. Probably he's asking the use of it.
So it is a tool. We can use this tool to study like protein-protein interactions, like what we studied in the cell culture system. So ULK1 and MNOSOD interaction we can study using bioinformatics tools. So many things can be done using bioinformatics tools.
Sir. Another question is from Dr. JP Ratha. The question is, if we expose an animal to stress, how the trade-off takes place between ULK1 and BRCA1 overexpression?
When we the animals, even when we do starvation, human do also starvations, there is autophagy activations. And ULK1 is the initiator. So definitely the ULK1 activation will be there, phosphorylation will be there, that leads to autophagism formations and autophagy inductions. And BRCA1 is a tumor suppressor gene. So whether it is related with ULK1, that I don't know.
So the next question is from Dr. Durga Prasad Panigrahi, and the question is, can we trigger autophagy to cure cancer?
Yes, this is the whole chapter we discussed that. So autophagy in cancer is context dependent. So we have to first check the status of autophagy in cancer, whether autophagy is protective or autophagy is lethal. Depending on it, either you can inhibit the autophagy or you can promote autophagy to treat the cancers. Like in the during cancer progressions, so there is a different inhibitor, mostly chloroquine. Hydroxychloroquine is used for cancer treatment along with the chemotherapy.
Okay, thank you, sir, for answering all the questions. I think we have another question. The same question, like the stress of the animal, and it is between ULK1 and BCL overexpression.
BCL2 inhibits autophagy. So when the BCL2 overexpressions, So there will be during stress, when you overexpress BCL2, it will inhibit autophagy. BCL2 interact with the Becklin 1, and in Becklin, as a result, there is inactivation of autophagy. Autophagy, there will be inhibition of autophagy when we overexpress BCL2.
Thank you, sir. Thank you for answering all the questions, and thank you for your nice presentation on the topic autophagy and its role in cancer. Now we will move to the next speaker.
Thank you.
Now I welcome Dr. Iswar Baitharu. Currently, he is serving as Assistant Professor in PG Department of Environmental Sciences, Sambalpur University. Dr. Baitharu has done his PhD at the Department of Neurobiology in Defense Institute of Physiology and Allied Sciences, DRDO, Delhi. He has worked as a research associate at Department of Biotechnology and Bioinformatics, JP University of Information Technology. He has also worked as a research scientist in Bill and Melinda Gates funded project as International Center for Genetic Engineering and Biotechnology, New Delhi. Worked as a research associate at Department of Neurobiology, Jawaharlal Nehru University, New Delhi. He has 24 publications in various journals like Neurobiology of Diseases, +1, Journal of Ethnopharmacology, Neurobiology of Diseases, High Altitude Medicine and Biology, Behavioral Brain Research, Neuroscience Letters, Cellular and Molecular Neurobiology, Brain Behavior and Immunity, and Journal of Neurochemistry. He has 388 Google Scholar citation. So now I invite Sir to present his talk on the topic effects of stress hormone and on stress-induced cognitive dysfunctions. Sir, please, now you can start your talk.
Hello, good morning, good afternoon, everybody. And thanks, Sagatika, for your nice introductions. I hope I am audible to everybody. Can anybody confirm?
Yes, sir, you're audible. Yeah. Yes, sir, you're audible.
So, first of all, I will extend my heartfelt thanks to Pragya Parimita for organizing such a beautiful webinar. And today I will present a topic. It's a kind of general topic like the role of stress hormone in stress-induced cognitive dysfunctions. Let me share my slide, please. Just coming to it. Is my slide visible to all? Hello.
Yes, it is visible.
Okay. So last few months has been very dramatic because of the corona pandemics, and this corona pandemics actually have taught us and defined what actually we mean by stress. Okay, so recently the WHO has released few press briefs where the WHO has surveyed across the globes and found the mental health of human beings living under COVID pandemics are actually deteriorating. And this is occurring because of the stress of COVID, uncertainty that are existing in COVID pandemic situations. So this is the the impact of COVID-19 on mental, neurological, and substance use services. And WHO also has released guidelines how to manage your mental health in COVID pandemic situations. So what exactly is that the existing COVID situation has created huge amount of stress, mental burden on the people's living. So many people people's lost their job. Some of them lost their life also. So this situation has created a kind of stressful conditions. So what exactly we mean by stress and how we can adapt to the stressful conditions in existing situations. So stress is basically we in our day-to-day life we are exposed to various kind of stress. Stress can be any kind of perturbance or disturbance that basically create strain in our body systems. Okay, so when any organism is getting exposed to stressful conditions, the simple things that happens is it is perceived by a particular region in the brain that we call the somatosensory cortex. So the neurons in the somatosensory cortex once get activated, it passes on the neuronal signals to the hypothalamus, and hypothalamus activations lead to the activation of the entire axis that we call it the hypothalamo-pituitary adrenal axis. So once this hypothalamo-pituitary adrenal axis is activated, a stress mediator is released that we call popularly as the stress hormone. So depending upon the concentration of the stress hormone released, there are appropriate physiological response and behavioral response occurs in the living systems, which enables them to adapt to the stressful conditions. But sometimes what happens, even though the organism is not under stress, but the stress hormone level goes up, or the organism is under stressful condition, but the required amount of stress hormone is not secreted. And these two conditions make the situation deleterious. So when such a situation arises, then the mechanisms that we called as the allostasis get activated. So allostasis basically is a homeostatic mechanism by which the body tries to maintain constancy in every physiological parameter. If this allostasis process failed, then wear and tear occur in the living organ systems, leading to the increase in the allostatic load, and allostatic load beyond certain limit make the organism susceptible. So if allostatic load become high, maladaptation begins. So in this COVID situation, this is one of the things that is happening. When maybe, but so depending upon the allostatic load, how much stress we can tolerate, that make the organism susceptible to the stressful condition, or he may get adapted to the stressful conditions without showing any malfunctioning in mental health. So in this entire story, the most important role is played by the stress hormone or the stress mediators. So what is that stress mediator actually? So stress mediators are actually one of the most important stress mediators is this stress hormone that is called as the corticosteroids. Okay, corticosteroids are basically secreted from a adrenal gland. Adrenal gland itself is a small bean-shaped gland situated just above the kidney. It's a suprarenal organ. And if you take a cross-section of it, you will find the two regions, visible region. One is the cortical part and the other one is the medullary part. And microscopic observations will show you the three different regions. Okay, so capsule, cortex, and medulla. And macroscopic observations will see, like in cortex region, you will find zona fasciculata, zona reticularis, and zona glomerulosa. And the corticosteroids is basically synthesized in these regions, cortical regions of the adrenal glands. So, and as you know, corticosteroids is the steroid hormones. The precursor is cholesterol. These cholesterol undergo several enzyme catalyzed steps, and it gets converted into corticosteroids. So the most important part is that corticosteroids is basically extra cerebral in origin. Means it is produced outside the brain, and it has to be transported to the brain to activate the HP axis. Okay, so, and as you know, the corticosteroid is lipid solubles. It is the steroid hormones. It cannot be transported easily through the circulatory systems. So it basically binds to carrier molecules that we call the corticosteroid binding globulins. So what are the factors that actually regulate the entry of corticosteroid to the brain? There are three important factors that actually decide how much of corticosteroid will enter to the brain. The first one is the 11 beta hydroxysteroid dehydrogenase. It's a pre-receptor metabolic enzyme, and this 11 beta hydroxysteroids dehydrogenase has two isoforms, HSD1 and HSD2. So what it do, it actually convert inactive form of corticosteroid into active forms. Okay, so HSD1 convert the inactive form, that is the dihydrocorticosteroid into corticosteroid, and beta HSD2 do the reverse. Okay, so if beta HSD1 is more, that means that indicates that the corticosteroid in active state is more. Okay, then the second thing is the multi-drug resistance phosphoglycoproteins. So excessive accumulation of corticosteroid in the brain, if it occurs, then that are actually pumped out of the brain by this multi-drug resistance phosphoglycoproteins. Okay, so and the corticosteroid is being transported through the blood to the brain by the corticosteroid binding globulins. That is the reason we call this as the static regulator. So actually, what happens? Whatever corticosteroid is secreted to the blood, it remains in bound form. Because so 95% of the total corticosteroid in blood remain in the bound forms, and the bound forms of the corticosteroids are not basically not available for signaling purpose. So the corticosteroid binding globulins actually regulates, if the person is under stressful conditions, an immediate corticosteroids release is required. That time the glucocorticoid, the CBG proteins expression decreases, or it increases the free hormone available for signaling purpose. So that is the reason we call this as the static regulator of corticosteroid. And this is the feedback loop. Feedback loop by which the corticosteroid itself regulate the through the feedback pathway. It can either stimulate the paraventricular nuclear cells, and it reduce the release of corticosteroid releasing hormones, and further the corticosteroid amount decreases. So it's like the known fact that everybody knows. Okay, so what exactly the corticosteroid do? The corticosteroid is like a double-edged sword. Means it is neuroprotective at some times, and it is neurodegenerative at the other point of time. So, and depending upon the concentrations, like you see, if the corticosteroid level is a physiological level or sub-physiological or slightly elevated levels, it provides neuroprotection. But chronic absence of corticosteroid, or if there is stress-related high concentrations for longer period of time, the same corticosteroid become neurotoxic. And there are several pathways by which it kills neurons. Okay. So now, if a person is under stress for longer period of times, the corticosteroid may remain elevated for longer period of times, and it may causes neurotoxicity. So this may be happening in the current situations of COVID pandemics. Now, if we come to the neuronal systems, now if you come to the memory or the cognitive functions, this is the limbic system. Limbic system is basically the most important system for cognitive functions. If you see this is the region where the hippocampus is there. So what is hippocampus? Hippocampus is the part of temporal lobe in the human brain, which otherwise known as the CPU of the human brain, or central processing unit of the human brains. It is because of its shape, we call that as hippocampus. It's look like the seahorse. If you see the morphological similarity, it's look like a seahorse. That is the region we call this as hippocampus. And if the morphologically you see, it is the hippocampus, and it is the dentate gyrus regions. Dentate gyrus regions where basically neurogenesis takes place, and this is the here hippocampal regions. Hippocampal regions is basically act as the processing unit of human brain. So now there are several pathways. There are pathways by which the sensory inputs, whatever we are getting, are processed through the hippocampus. Now you see this from this is called as the perforant pathway. So whatever the information that comes from the sensory organs, it passed through the entorhinal cortex and reached to the dentate gyrus. So here is one synapse. Okay, synapse means the connection between two neurons. Okay, so this is the perforant pathway from entorhinal cortex. It is reaching to the dentate gyrus regions of the human brain, hippocampus, and then from dentate gyrus it is going to the CA3 pyramidal neurons through the Mossy fibers. Okay, so this is another neuronal tract. So there is the second synapse that is taking place here, and then from these CA3 regions it is going to the CA1 regions of the hippocampus through this scaphocollateral pathway. So synapse 1, synapse 2, synapse 3, that is the region we call this as the trisynaptic circuit. Now this synapse, in between these two synapses, presynaptic neurons and the postsynaptic neurons, there are several different types of neurotransmitters that are released. Okay, so as you know, there are three different types of neurotransmitters altogether. One is excitatory neurotransmitter, inhibitory neurotransmitter, and modulatory neurotransmitters. So depending upon what types of neurotransmitters is released from the afferent neurons, the subsequent your cognitive function will be displayed upon. Okay, so after passing through the trisynaptic circuit, the information will go to the cortex where it will be stored for retrieval process. So if information pass through all the three synaptic pathway, later and it is stored in the form of information in the brain. So memory is stored, that we call as the memory. Okay, so how this memory is formed, so how we are able to remember things. Okay, so as you know, memory has three different steps. The step one is acquisition of information, second is your consolidation, and then third one is the retrievals. So once whatever information that you are getting, you are acquiring. Once information is acquired, it is consolidated. Okay, once consolidation takes finished, then it becomes stored, and later on it can be retrieved as and when the person is required to remember things. Okay, so information is basically stored in the form of synapse. Okay, it is the whatever memory that are stored in the brain, it is stored in the form of synapse. So if you see, this memory is a special case of general biological phenomenon of neuronal plasticity, and you can see this if you see this is the millisecond, then second, then minute, then hours. So what is happening here? There is once information pass through a synapse, the synaptic strength is increasing. You see the pathways, the calcium release and the subsequent genetic process is taking place, and it is released to the increasing the synaptic strength between the presynaptic and the postsynaptic pathway. So all synapses cannot be stored for long. So if, let's say, any information that are very repetitive in nature, that will be converted into long term, means short term to long term memory. Okay, so learning induces cellular and molecular changes that facilitate communication among neurons and are fundamental for memory storage. And as I have already told, if learning brings significant synaptic strength, it can be converted into long-term memory. Okay, so what is happening? Most of these synapses that are formed when we are coming across many things, it is getting faded away with time. That we call as the forgetfulness. So forgetting is part of memorization of things. So if somebody is not able to forget anything, the memory or the cognitive dysfunctions will start from that. Okay, because too much of information, too much of synapse formations can reduce new synapse formations, and that could be deleterious for the organism to remember new memory, formation of new memory. Okay. So what is happening? If something is coming repeatedly, then what will happen? Whatever stimulus is coming, it will be first converted into short-term memory, and later it will be consolidated through the long-term memory. But the consolidation process require expression of number of gene, transcription and formation of proteins. Okay, so if it's a long process, consolidation require time. So short-term memory, if it is persist for longer period of time, it will be converted into long-term memory, and because it requires gene expressions, if any inhibitors is there, if suppose there is mRNA half-life is less or the protein is unstable because of the presence of inhibitors or any other molecules, if the consolidation process is disturbed, that means the long-term memory cannot be formed. So, and whatever memory is there, we can have active memory at a time. So active memory is basically reconsolidated form of memory that basically come into action every time. Okay, so this is the mechanism of short-term and long-term memory here. You will see this is the presynaptic neurons and this is the postsynaptic neurons, synaptic cleft, and the neurotransmitter released from the presynaptic neurons to the neuronal synaptic cleft, and where there is two important receptors. One is the AMPA receptor, alpha AMPA receptor, and the NMDA receptor. This NMDA receptor is basically a calcium channel. Okay, so what is happening is it releases calcium, binds to the calcium calvulin pathway, and depending upon this, there will be NOS signaling and signaling through the PKC, CAM kinase 2, tyrosine kinase, calcineurins, which lead to the formation of short-term memory. Now, if it occurs, if this is to be converted into a long-term memory, then the adenyl cyclase pathway get activated, and it requires modulatory inputs. Now, this conversion will take place the another signaling pathway that we call as the protein kinase A, PKA pathway, and this is facilitated by the expression of several genes through the Krebs signaling pathway. So I'm not going to detail of it. So what is the relationship between stress hormone and memory? If you see this graph, this is a U-shaped relationship. Means this is the working memory errors and the concentration of corticosteroid in microgram per deciliter of blood. See this, if the corticosteroid concentration is less, the working memory is good initially. And here the working memory error is less, okay. And it is, the concentration is less, working memory. And if you see this, there is higher concentration of corticosteroid, the working memory is increasing. So here from this, so very less amount of corticosteroid is also having more working memory errors. Very high concentration of corticosteroid also having higher working memory errors. That means there is an optimal concentration which is required for formation of memory. So optimal, here you can see this, it's come around 20 microgram to 50 microgram per deciliters, where the memory working memory error is very less. That means this is the most desirable concentration of corticosteroid that is required for memory formations. Okay. Now what I did here during my PhD, I took hypobaric hypoxia as the model stress model. And this why it is relevant to this present scenario of COVID-19. So you might have heard the earlier speakers where they have shown COVID-19 itself is a respiratory illness. It is caused by the viral infections. So what is happening is disrupting the oxygen diffusion from the lungs to the blood cells. So indirectly it is causing hypoxia in the tissue level, at the tissue level. Okay. So if I take these models to an environmental stress like hypobaric hypoxia, the same thing is happening in this case also. So if you go up, what is happening? The partial pressure of oxygen is decreasing with time. So up above around 5,500 feet, it's very difficult for the organisms to have appropriate amount of oxygen in the blood, since the organism cannot survive. So the high altitude regions begins when we climb up around 1,500 meter and above. So what is happening in this case? It has been observed when a person gets exposed to hypobaric hypoxia, there is decline in physical efficiency, high altitude maladies are seen, then because of the extreme colds, hypothermia may be seen, and there are several other things that are found in the high altitude regions. But we are interested in this part. This is the impairment of cognitive functions at hypobaric under hypobaric hypoxic condition. So I've just be moving faster. So under this background, we try to know what kind of effect the corticosteroids might be having. So what we did, we studied the temporal variation in corticosteroids and their receptors, and we also tried to know what are the factors that affect the availability of corticosteroid in the brain. Okay, so here I'm not showing the bioenergetics part. So what we did is we took space dolly rat, and we randomly divided these rats into 5 groups: normoxia, 3 days hypoxia, 7 days hypoxia, 14 days hypoxia, and 21 days hypoxia. And then we studied the different parameters like level of corticosteroid by SPLC. We also studied the morphological alterations and the expression of receptor and transporter. So how we did hypoxic exposure? This is the animal decompression chamber where we can maintain the conditions that are available at high altitude regions around 25,000 feet above. Whatever partial pressure is there, that partial pressure can be maintained inside this animal decompression chambers that are there, that were available in the high altitude lab at DRDO. Okay, so we estimated corticosteroid level both in plasma as well as in tissue. Okay, you can see this. There was significant increase in the corticosteroid level in the hippocampal tissue when the animal was exposed to hypoxia for 7 days. Okay, both in your tissue level as well as in the plasma levels. Okay, so and the earlier reports from our laboratory, it has shown on the 7-day hypoxic exposure, there was significant increase in the neurodegeneration in the hippocampal cells. Okay, so we wanted to correlate between these two, whether the hippocampal neuronal damage that are seen at the 7th day of hypoxic exposure, whether they are because of the excessive corticosteroids or because of some other factors. Okay, so what we did, we estimated the factors that are actually regulating the corticosteroid levels in the brain. So you can see here we studied the beta HSD1 and MDRPG. Okay, so what is happening here, you can see on the 7th day hypoxic exposure, beta HSD1 is more. Beta HSD1 is the enzyme which basically converts the inactive form of corticosteroid into its active form. Okay, so that means whatever corticosteroids is there in the brain, they are in active state. Now you can see this beta MDR1 PG, MDR1 phosphoglycoproteins. Here the expression on the 7th day is very less. That means the accumulation is occurring because of the inability of the phosphoglycoprotein to pump out those excessive quantity of corticosteroids that are accumulated in the brain regions. Okay, so now you can say like the whatever fraction of corticosteroids is there in the brain on the 7th day of hypoxic exposure, they are the active fraction of corticosteroids. Okay, now we also studied the expression. Now the corticosteroid will bind to the receptors. It has been reported that there are 2 different types of receptors that are found in the brain. One is the for corticosteroids. Corticosteroid can have 2 receptors. One is the glucocorticoid receptors that we call as the low affinity receptor, and the other one is the mineralocorticoid receptor, or called as the high affinity receptor. So when the corticosteroid is the lesser quantity, basically it remains in bound form with the mineralocorticoid receptor. Okay, but when organism is exposed to a stressful condition and the corticosteroid level increases, all the mineralocorticoid receptors get saturated, then the extra amount of corticosteroid will bind to the glucocorticoid receptors. It has been reported that the transient binding of corticosteroid to the glucocorticoid receptors are beneficial. Okay, it enhances the performance of the person. But if this binding become chronic and it's occur for longer period of times, then it become deleterious. Okay, so here you can see on the 7th day, the glucocorticoid receptor expression is maximum. Okay, but the mineralocorticoid receptor also is there. Okay, but if you see the 14th day data, it is more interesting. In the 14th day data, you can see the glucocorticoid receptor is decreasing, but the mineralocorticoid receptor expression is increasing. That means there is shift in the GR by MR ratio, and the 14th that means what? The reduction in the glucocorticoid receptor and in the mineralocorticoid receptor indicates the onset of another process that we call the acclimatization process. Okay, acclimatization process is the process by which the organism started adapting to the stressful conditions. Okay. Now what we did, we tried to manipulate the corticosteroid level in the brain. Okay, and how we did it? We did it by taking another molecules that we call as metirapone. Okay, and the metirapones was administered 50 milligram per kg body weights, and the other parameters were studied. We estimated after metirapone, we actually basically we choose the 7th day hypoxia only because we observed maximum neurodegeneration on the 7th day only. Okay, so what we did, we administered metirapone to the rat, and the metirapone is a very interesting molecules because it only inhibits the stress-induced elevation of corticosteroid, but it doesn't inhibits the basal corticosteroid level. Okay, because it is a competitive inhibitor of 3-beta hydroxy steroid dehydrogenase enzymes, which is responsible for synthesis of corticosteroid. So because it is competitive, it does not completely inhibits the synthesis of corticosteroid in the adrenal glands. It only reduces the corticosteroid to certain levels. Okay, so we confirms whether this is happening. So you can see this on the 7-day hypoxia and vehicle treated groups, the corticosteroid is very high, both in plasma as well as corticosteroid. But when they are we administered metirapone, okay, so the concentration decreases. Okay, it is just above the normoxia level, but it is significantly decreased compared to that of the hypoxic group. That means the metirapone is maintaining a slightly elevated corticosteroid level in the brain under hypoxic condition. Now we also studied the oxidative stress markers and found the metirapone is basically reducing the oxidative stress. You can see this, the loss generation is decreasing. It is increasing significantly the glutathione reduced glutathione levels, and basically that is the reason there is a shift between the generation of corticosteroid free radicals and removal of free radical from the neuronal systems. Okay, so then we studied the hippocampal region. This is the CA3 regions of the rat, where we studied how whether neurodegeneration is occurring when we are subjecting metirapone to the rats. You can see here there is significant increase in the pycnotic cells in the CA3 regions. This is the pyramidal neurons. So there is significant increase in the pycnosis when the rats are exposed to hypoxia. Here you can see this. But when we administered metirapones, there is significant decrease in the pycnotic cells. But this is that means what the metirapone is providing protection to the neurons at the CA3 regions when metirapone is administered under hypoxic conditions. Okay, but whether the metirapone is directly doing this or it is doing by reducing the corticosteroid level, because metirapone can also act as a neuroprotective agent. By it can have several other signaling pathways can reduce the pycnosis in the CA3 region. So what we did, we simultaneously inject metirapone as well as the corticosteroids. That means if corticosteroids remain elevated, even if there is metirapones, again you can see there was very significant increase in the pycnosis in the CA3 regions of the rat. That indicates that the metirapones, the neuroprotection that are provided by the metirapones is basically corticosteroid dependent, and it is not independent of corticosteroid in the brain. Now we studied the apoptotic cells, also caspase-3 genes expressions, and you can see metirapone has significantly decreased the caspase-3 expression in the hippocampal region, CA3 regions basically. Then we tested the memory status of this rat. You can see here, this is the Morris Watermaid's data. Here we have, this is the pool, and we have divided into 3 quadrants, and this is the target quadrants actually. Here we have trained the rat to find out the submerged platforms in the pool, and we have trained it in the normoxic conditions. When we are removing this platform from the pool and we are putting the rat is only traveling in the quadrants where the platform was located. That means it can remember that there was a platforms in the pool in this quadrant only. The memory is intact. Okay, but in normoxic condition with metirapone, you can see it is traveling only in the region where the metirapone was there. But in case of hypoxia treated group without any drugs, you can see it is because the memory is getting impaired. It is moving all through the quadrants, all through the pools, and it is actually uh, showing very less time in the target quadrants. But when metriapon is treated, this again the restoration of memory is being seen here. So what we did here is we studied the number of entries to the target quadrant. There is significant increase in the number of entries to the target quadrant. Then the time spent in the target quadrant also has increased. And we in this case you see here the latency to reach to the after you release the rat, how long it takes to reach the platforms. You can see here Narmoxia and Narmoxia plus vehicles, very small times it is taking 6 seconds or something. But in hypoxic group you can see because there is memory impairment, it is taking more longer times. Sometimes it also it can go time out after 60 seconds till for 60 seconds also it is not able to reach to the target point. So but when the metriapon was administered, there was significant decrease in the latency in both the cases. Okay. So this is our conclusions that the metriapons, that means what? It's metriapon is just the symbolic one. If you can reduce the corticosteroid level in the brain, manipulate the corticosteroid level in the brain under hypoxic conditions, under stressful conditions, you can increase the memory functions, and this memory functions increase is basically dependent on the glucocorticoid receptors and mineralocorticoid receptors. So there's a fine balance between MR and GR bindings. So MR binding is protective and GR binding is degenerative. Okay, so you can manipulate and make the binding of corticosteroid transient to GR. We can enhance the memory functions and cognitive functions. So this is the findings of this. Thank you very much for your patience sharing. If you have any questions, please ask. Hello.
Thank you, sir. Now I request the participants to put their question in the chat box. So the first question is from Dr. J.P. Rath. What happens in Alzheimer, whether there is any role of AMDA receptor?
Which receptor?
AMDA receptor.
AMPA.
Yes, sir.
AMPA receptor.
Yes, sir.
AMPA or NMDA? EMPA.
AMPA, I think, sir.
Okay. See, AMPA receptors actually basically Alzheimer's is a neurodegenerative disease. Basically, it's a progressive neurodegenerative disease. It is occurring because of the accumulation of a misfolded protein that we call it the beta amyloids. Okay, so beta amyloids signaling is something little bit different. That means what? It's basically it goes through the necrotic pathway. Because of the excessive accumulation of beta amyloid proteins, the neurons get damaged. Okay, so but there are several papers which say there is the role of AMPA receptors because see, in case of Alzheimer's also there is loss of memory. Okay, and the memory formations is basically dependent upon this trisynaptic pathway. So if any neurodegenerations that occurs in the trisynaptic pathway, it will just disrupt the processing of informations in these trisynaptic circuits. Okay, so let's say if the amyloid proteins accumulated in the CH regions, it will kill the pyramidal neurons in the CH regions. Okay, thereby it disconnect the tract. But how the signaling take place, whether the AMPA is receptor, there are several papers. It says the AMPA also is receptor involved in this case. Okay, and the research is going ongoing one. Okay, AMPA receptor has involved in many other signaling pathways also. I think I have answered these questions. Any other questions,
Yes,
please?
sir. The next question is from Sweta Praharaj. Why do different animals react differently to a same kind of stress?
Okay, different organisms.
Yes, sir.
Okay. Even the same organisms at different stress conditions, they respond differently. Okay, so
Yes.
behavior is actually behavior is very complex process. If see, behavior is the phenotypic response of an organism. And like you see in case of your plant system, the color of the flower, just the phenotypes of some of the genetic constitutions that are there in the background. Okay, so because it is multifactorial in nature, because see, all organisms have their own unique behavioral pattern. As if you see the rat, even the mouse have different behavioral patterns. Okay, and it's very difficult to study the behavioral response of rat. You see, in human beings, if a person is crying, you can know from his facial expression. If person is very in happy modes, you can know it from the facial expression. But you cannot do the same things with the rat models. It's the same all the times. Okay, so whatever behavioral assessment that we are doing, we basically have several different behavioral measures that are used to assess the state of mind of the rat. Okay, so if you are asking me whether the rat is behaving this way, the mouse will be behaving the same way or not, so that will be another different things. Okay, even the we have basically in neuroscience, the basic informations were generated in the Aplasia model. Okay, it's mollusk models. It was used by the father of neuro memory formations, that E.R. Kendall. E.R. Kendall was the father who actually discovered the mechanism of memory formations. He was not working on mammalian models. He was working on Aplasia models, it's molluscan models. Okay, but to assess the behavior of the molluscan, you cannot apply the same tools that are applied for rat models. Okay, so because the behavioral pattern is different. Okay, so that is the reason. So one sentence answer is like, behavior is a multifactorial process, and each and every organism have their own behavioral pattern. Okay, that is the reasons. Even this stress remaining the same, organism will behave differently. Okay. Any other questions?
Yes, sir. The next question is how people in high altitude region adapt to hypobaric or hypoxic condition.
Yeah, this is a very good question actually. See, I was using a terms acclimatizations during my presentations. Okay, so the person who are native to high altitude, those peoples are basically they have some physiological advantage over the people who are living in lowland. Okay, so let's say persons who, like us, we are living in lowland regions. If we are migrating to the high altitude regions, to adapt to the high altitudes conditions, our body must modify itself. The physiological process must get upgraded with respect to the conditions existing in high altitude regions. But those people who are there, they are the native to high altitude regions. They have all the physiological parameters at par with the existing surrounding conditions. That is the regions in DRDO. For high altitude regions, we basically employ people who are native to that part, the Gurkha battalion. These peoples, Nepalian peoples, they are basically preferred over the peoples who are working, living in the base regions. Okay, because one parameter just I am telling you, this is with respect to the oxygen concentration. In high altitude regions, the partial pressure of oxygen is less. That make us very difficult for us to adapt. And the other things are there, the low temperature, extreme UV condition, and other things are there. I'm just focusing on UV, the partial pressure of oxygen. You see, so what is happening? The oxygens will be carried from lungs to the different parts of the body through hemoglobin. Okay, if you estimate the hemoglobin concentration in the people, those are native to high altitude region, you will find higher quantity of hemoglobin in their blood. It's kind of a polycythemic condition that are existing there. Okay, compared to that, if you compare with that of the people living in lowland. Okay, so if you are migrating from lowland to high altitude regions, suddenly your hemoglobin concentration will increase. Are you getting my point? So because their genetic makeup has been such that they are quite adapted to the high altitude conditions. Okay, so they are having advantage over us. Okay, they are basically having advantage over us. There are many other morphological parameters that are there, which are unique to high altitude regions, and they are having that. Okay. Any other question, please?
Thank you, sir, for answering all the questions nicely.
Thank you.
I think there are no questions.
Okay.
So
Thank you
now
very
I
much.
request, yes, sir. Thank you so much for your talk. Now I request Dr. Pragya Paramita Naik to present the vote of thanks.
Good afternoon, all. As all good things come to an end in life, so is the webinar. On behalf of PG Department of Zoology, Vikramdev Autonomous College, I take this opportunity to propose the vote of thanks to those who have directly or indirectly contributed to this webinar on current trends in life science research organized by our institute. At the outset, I thank our keynote speaker and resource person, Dr. Sujit Pujari sir, for readily accepting our invitation and giving his kind consent to enlighten us with his knowledge pool. Moreover, we are thankful to our esteemed resource person, Dr. Sujit Kumar Bhutia sir, Dr. Santosh Kumar Panda sir, Dr. Sanjay Kumar Sahu sir, and Dr. Iswar Bhattaru sir for widening our knowledge on respective fields. Today's webinar was full of knowledge and interesting things. It gave us deep insights into the topic and also revealed some interesting facts. The point where Dr. Sujit Pujari sir discussed about the SARS-CoV-2, the pandemic of 21st century, was really very informative. The talk by Dr. Santosh Panda sir on coronavirus, a challenge to immunologists, gave us new insight into the challenge to be addressed by immunologists to fight coronavirus. Furthermore, we were able to learn various aspects of complement biology from Dr. Sanjay Sahu sir. The talk presented by Dr. Bhutia on autophagy in cancer helped us to understand the dual role of autophagy through both tumor promoter and anti-tumor function. Finally, the talk of cognitive functions and stress response given by Dr. Bhattaru was thought-provoking. I am pretty sure the precious knowledge that all the eminent speakers that they have given through their talk will definitely help us in our studies and future. Once again, I would like to thank Dr. Sujit Pujari sir, Dr. Sujit Bhutia sir, Dr. Santosh Punda sir, and Dr. Sanjay Kumar Sahu and Dr. Iswar Patharu sir for taking out time from their busy schedule and share their experience and knowledge in respective fields. I am especially thankful to the speaker from abroad from their kind consideration for readily accepting our invitation despite the time constraint because of different time zone in India and USA. It has been our pleasure to host all the participants of the webinar. The participants were very enthusiastic and I am thankful for them coming here to attend the webinar. I would also like to thank our principal, Dr. Gopal Haldar sir, for giving permission to organizing the webinar session to conduct it. I want to thank all the technical supports department staff for their sincere help. An event of this dimension cannot happen overnight and needs meticulous planning and execution and an eye for detail. I cannot thank each one enough for their involvement they have shown and the willingness they have expressed to take on the completion of the work beyond their comfort zone. I'm also thankful for the administration in general for rendering us with all possible logistic support towards organizing this event and all the plenary speakers and delegates for their enthusiastic participation. I would like to extend my special thanks to my colleagues, Dr. Ratnakar Parida, Ms. Swagatika Padingrahi, and I would also like to extend my gratitude to Dr. Lakshman Patra sir and Mr. Gyanaranjan Mahanti sir and Dr. Sagarika Mishra sir for giving their moral and technical support during the webinar session. I'm also giving my special thanks to organizing committee, teaching and non-teaching staff for their unflinching support and coordination. Our heartfelt thanks to all the participants. With these warm words and a kind message, we move forward to the end of today's webinar. Thank you all.